So I think we will start, and I think Dr. Sakai will join us later. I can't see him. Okay. Okay, wonderful. So yeah, so we are going to start with a few words from Dr. Jake Bartlett, Professor of Surgery and is Vice Chair of Surgery Research, and also the Director of Research in the Division of Orthopedics. Dr. Bartlett, please. And Nico.
Okay, so thank you very much for giving me the privilege of opening the research day for 2022. For the students who are in their first year, you know, you've reached nearly the end of the academic year, so we hope you've enjoyed the past 8-9 months with us, and you know, you agree that you made a great decision. For those of you who are about to be moving on, congratulations and good luck. You've seen from the video, you know, what a fantastic job the organization committee has done of putting together a fantastic program for you. So my main job now is to thank them very much for all of their hard work over the past weeks, working as usual with incredible difficulty and uncertainty. Would it be in person? Would it be hybrid? Would it be whatever? So in particular, then we'd like to thank Matthew Manarino, Ida Derish, Oliver Martinez, Micha Hun, Sharon Turner, Michael Grant, Laura Epure, and of course, we wouldn't be anywhere without Professor Mwali, Graduate Program Director and Director of the Research Day today. You will see that we have an amazing selection of international, national, and local keynote speakers today, and I'd like to thank them all very much for giving up their time, evenings, mornings, for joining us today. Professor Daisuke Sakai, Professor Jason Harley, and Professor Rita Kandel. So welcome everybody, thank you once again, and I'll pass you over to Professor Mwali.
Thank you very much, Dr. Bartlett. It is my great pleasure to introduce the first keynote speaker, Professor Daisuke Sakai from Tokai University School of Medicine in Kanagawa, Japan. I've known Dr. Sakai for more than 20 years now. Wonderful person and great scientist. He is an associate professor in the Department of Orthopedic Surgery. His clinical interest is in the pathology of degenerative spine disease and management of spinal deformity, which includes adolescent idiopathic scoliosis, other spinal deformity and functional rehabilitation. So he also serves as the head of the Intervertebral Disc Research Unit, which studies the biology, homeostasis of intervertebral disc cells. And also they look at stem cells. There were some really wonderful studies which were published in Nature. Dr. Sakai has given over 310 presentations, invited lectures and seminars in universities, professional societies, international conferences. For example, he was an invited lecturer from 2004, 2010, and 2015 at the Gordon Research Conference on musculoskeletal biology, bioengineering, and intervertebral disc biology. So he has won a lot of awards and large national grants. And among them, one of the awards is he was the winner of the 2014 Research Achievement Award for the Japanese Orthopedic Association and 2018 Beton Run Research Award from the EO Foundation. He has published over 160 papers. He chairs he has served as a chair for the International Committee of the Japanese Orthopedic Association and chair of the International Committee of the Japanese Scoliosis Society. He also serves as a co-editor of the Journal of Orthopedic Research Spine. So this is basically an official journal of the Orthopedic Research Society. So he will be research committee chair of the Asia Spine Research Society. He will also be the secretary starting from 2023 to 2027 for the ISOS, or International Society for the Study of Lumbar Spine. The title of the talk is Clinical Development of Allogenic or iPS cell-derived Tie2 positive nucleus pulposa progenitor cell product for low back pain. Dr. Sakai, please.
Thank you, Dr. Moali. And can you hear me well?
Yes.
Okay. So I would like to thank the organizers and especially Dr. Bartlett and Dr. Moali for having me here today to speak in front of many young scientists on musculoskeletal biology. So today I would like to speak a little bit about our challenges for clinical development of allogenic or iPS cell-derived Tie2 positive nucleus pulposus progenitor cell products for low back pain. Here are my disclosures. So low back pain is a very big burden on societal and economic burden on modern societies. And there is no absolute or ultimate curative treatment associated with this degeneration. So we studied the biology of the intervertebral disc, as Dr. Moali is one of the frontiers in this area as well. The intervertebral disc is a largest vascular tissue, which means that it is left away from the vasculature and nutrition and cells that can migrate into the disc to repopulate and regenerate the tissue itself. So it has a poor regenerative capacity. Postnatal vascular regression occurs within the developmental phase, and you can see that the vasculature is suppressed, which are coming to the end plate but are not penetrating into the disc, which is a a big characteristic of the nucleus pulposus or the intervertebral disc itself. The normal intervertebral disc extracellular matrix has been shown to prohibit angiogenesis. We do not really actually know what prohibits the angiogenesis itself, but it seems that the aggrecan and the sugar itself can prohibit angiogenesis with mechanism of anti-angiogenesis derived from the cells in the ECM. Pain is sensitized. It's not penetrating into the disc itself, but it has afferent nerve. It sprouts into the outside layer, which is the outer annulus, which connects to the DRG, which sensitizes the pain CGRP related sensory fibers. When the disc degeneration occurs, these vasculature and the nerves will ingross into the disc in response to the inflammatory agents which are secreted by the central nucleus pulposus cells, and will let us know that there is a structural damage or whatever that's going on that might result into structural deformity or structural catabolic mechanisms. So the regenerative potential decreases with aging, and genetic predisposition is also have a big effect. But mechanical loading and trauma, which means that everyday use in the modern society will, in some degree, accelerate the regenerative potential, decelerate the regenerative potential of the intervertebral disc. We know that the cells in the disc decreases with the number of cells decreases with aging, and other biomechanical or sources of outside phenomenon, which can lead to degenerated and accelerated degeneration. When that occurs, the biomechanical properties of the intervertebral disc prolapse and sometimes causes a lumbar disc herniation like this in the slide, which can directly affect the neural elements, which can lead to acute pain. But on the other hand, chronic pain that can result into after 3 months or so, if the patient is not recovering with pain, it can lead to chronic pain, which may be one of the causes of sustained inflammation and neural and vasculature has invaded within the avascular niche of the intervertebral disc. So the mechanism of dyscogenic low back pain is thought to originate from this effect of structural damage and inflammation, which can elicit innervation of the deep nerve ingrowth and upregulation of neuropeptides. And then the structural damages causes hypermobility, and then the acceleration of the degeneration process will start to progress in a faster speed. And then the sustained low back pain will cause psychosocial factors, etc., etc. And then we would have to do infusion surgery or extensive rehabilitation, or treatment options are limited though. The source of chronic low back pain, which lasts more than 3 months, is thought to originate from different pathology or different organs. One study has shown that the disc may be 40%, maybe up to 40% of the chronic low back pain may be originated from the disc, and compared to the facet joints and sacroiliac joints. Disc degeneration and disc herniation also affects a large population of the modern society, and which is a big societal problem. You can see here that the 4-5 disc has a prolapsed disc with a 3-4 disc with mild disc degeneration, which might lead to accelerated degeneration if the intervertebral disc is not taken care in terms of biomechanical influences and so forth. Chronic low back pain patients may seem to have this kind of imaging characteristics with high intensity zone, which is the white posterior portion of the annulus fibrosis, showing that there is an inflammatory response of the disc, although the patient may not have created disc herniation. So we need to have a curative treatment source, which we do not have that yet. And our community has looked into various treatment options using many animal models. And modeling of the disc degeneration has been performed using several methods like stab, puncture, nucleotomy, whatever. But the ultimate model that models the human intervertebral disc is still lacking. And we have enough animal data to support that. We've been working on injection of active cells to the intervertebral disc. And 50 or more modeling studies has been published showing that structural and imaging and biochemical changes can be reversed using cell injection when the disc degeneration is not too much far progressive. So yes, in the long term, this may be happening, but patients come to us for immediate pain relief. So these animal studies has limitation in that sense. Clinical investigation is truly needed in order to answer these questions, whether this type of new technology can be applied. So we and the others have shown that stem cell transplantation into the disc can reverse the progress of disc degeneration with active cells taking over the degenerative process and start to produce proteoglycans, ECM factors, any other factors that are needed to promote or halt the degenerative process with paracrine systems and ECM production from the native cells as well. So we tested in a human trial where if activated nucleus pulposus cell transplantation is safe, can be safely advised, we injected them into the adjacent segment disc disease, which was only the pathology allowed from the national authority. And you can see that the 20-year-old kendo athlete who failed for low back pain and had a 3-4 disc, and 4-5 disc was unstable, so it was fused, taking out the cells from the 4-5 and injected the cells to the upper level. Screws were removed, and you can see that after 6.5 years, the disc looks quite healthy. And she has been back to the normal athletics, and she has not had low back pain since. Cell therapy trials are increasing along the globe. There is already phase 2-3 out. Various cells and different modalities are used to investigate the potential of this idea. There is already a RCT, randomized clinical trial with mesenchymal stem cell, which was not as highly potentialized as we expected, but barely showed significance compared to the placebo. So there are lots of things that needs to be investigated still to maximize the effect of this treatment. And there's lots of placebo effect in this type of clinical trials. And what is the appropriate control? What's the definitive effective indications, and identifying the effective cell types for different conditions, etc. So in Japan, we have started these 3 generations of cell products. One is, the first generation was a bulk allogenic product from the venture company in the United States. They get the cells from the fresh cadavers and produce bioactive discospheres. And we did an animal study using their cells and promoted to clinical trials. But at that stage, we thought that this idea of this product could be more upgraded if we were to able to isolate progenitor cells from young donors. So the second generation allogenic type 2 positive cell product was developed. Well, it's still in development, and we are getting ready for next clinical trial using the second generation allogenic product. And third, if we were to able to create type 2 positive NP cell product from iPS cells, we are free from immune system, or we can have, HLA matched iPS cells. So we are in collaboration with CYRA, which is Professor Yamanaka's institute, and developing that direction. So the first generation comes from the USA. This is the Discosphere product. You may have known them in the news. We did a large animal study using their cells, which showed that the cells off the shelf, out from the freezer, and then inject them directly into the disc can have some kind of a positive effect. So we proceeded into clinical trial, and recently there was a good result in terms of halfway through the clinical trial in the US study, and we're still waiting for the results of the phase 1/2 study for the Japanese study, which may open the door for the first cell therapeutic application to the Japanese market as well. So for the second generation, we wanted to focus more on science, and the science, which means that the niche of the intervertebral disc is a characteristics of the cells. And if you want to regenerate a tissue, you need to really focus on the local niche and what controls the cells and their behavior. We discovered a set of cell surface markers in 2012, identifying an NP nucleus pulposus progenitor cells, controlled by the cell surface markers Ty2 and GD2. Ty2 positive NP cells have high proliferative capacity, and the differentiation has a hierarchical marker change, with Ty2 being the most primitive, and CD24 meaning having CD24 positivity, meaning the cells are really matured. Clonal characterization of these cells were performed with single cells, which showing proliferative capacity more than the bone marrow mesenchymal stem cell through first and primary and secondary clonal cultures, and they are clonally multi-differentiatable if you choose the right clones. The cell transplantation of these progenitor cells had sustained survival compared to CD24 positive NP cells, which are already committed adult NP cells. And these type 2 positivity are related to aging, meaning that after 30 or 40 years of age, in a lumbar disc herniated patients, you can see that there is a drastic drop of type 2 positivity in the cells. And then the colony formation units are also decreasing with aging, meaning that the avascular tissue has maybe in the lower region of the lumbar spine, the disc has the capacity to survive more than 30, 40 years, but after that depends on how much you have saved your stem cells. And decrease of, so the TIE2 and GD2 are identified as markers to define nucleus pulposus stem progenitor cells. And TIE2 angular niche accounts for NP cell progenitors, and exhaustion of these cells with aging can lead to disc degeneration. So after we published this, these data were reproduced by other collaborators and other laboratories in the Switzerland and China and other areas of the parts of the world, and have shown that type 2 positivity is one of the markers that can identify the NP-specific progenitor cells. We have published a review in Nature Review Rheumatology recently on several cell types that populate the disc and account for homeostasis of the disc degeneration process. And if type 2 positivity is a dogma or not is our main focus in terms of developing these types of cellular products, and also curation of the intervertebral disc degeneration. Type 2 positivity cells are one of the markers to show spheroid-like clonal proliferation. And we have shown that some of the type 2 ANG1 pathway and its stimulation can upregulate the effect of these type 2 progenitor cells, so that we can manage to create and develop cell products for many patients from a small donor of tissue. If we use certain culture condition and also depending on the donor, we can create maybe around 1,000 to 5,000 units of lots of type 2 positive cells from just 5 grams of disc material from young donors. And they can be functional and can perform to create extracellular matrix that are characteristics of the nucleus pulposus cells. And in vivo, they have shown high potentiated regenerative capacity compared to other cell sources. So after doing that, we have collaborated with pharmaceutical company and licensed out this idea and the patent in order to flow a manufacturer flow. So we are still in the process of getting ready for clinical trial using these type 2 positive NP progenitor cells for intervertebral disc degeneration, which going into commercialization is a big hurdle. And we learned a lot, but it's certainly coming, and lots of different regulatory responses were needed. Lastly, I would like to touch on some of the newest data on iPS-derived tied to progenitor cells. There's in the literature, there's a lot of recently a lot of papers on generation of or induction of NP cell-like cells, mimicking NP cells from MSCs or iPS cells or whatever. But generally, they are involving general chondrogenic factors only. We need to strategize these conditions to specifically more induce NP cells. So we wanted to define what are the key transcription factors that were needed to induce human cells into induced NP cells. So we narrowed down the transcription factors using different pathways. We narrowed down the transcription factors using microarray, iPS cell interference, siRNA assay, and upregulation and reprogramming assay. So this is a quite complicated step, but we compared from other cell types and defined what transcription factors were really specific to NP cells using microarray. And we used this differentiation mechanism in that sense. And you can see this, I'm not going deeply into the process, but by using these information, we were able to narrow down the transcription factors that were highly expressed in the nucleus pulposus compared to other types of cells, and narrowed them down into 200. And then we induced them into iPS cells with an Yamanaka factor, which makes NP cells into iPS cells, a reverse transduction pathway. We put in Yamanaka factors into the NP cells and made iPS cells. But if you have a very strong characteristics of NP cells mixed with the Yamanaka cells, that would interfere with the iPS cell generation. And if we were to completely deny iPS cell induction using a strong transcription factor, that would mean that that transcription factor is very important for NP cell induction. So this is the result. You can see that there was a complete interference in 8 or 9 transcription factors, including the BRCA-T. And then there's around 20 strongly interfering transcription factor. And we used this 80 or 90 so transcription factors to the next stage and did a silencing assay to see if this strongly silenced NP cell characteristics. And then we used the 25 transcription factors for direct reprogramming, and then induced NP cells into the from mesenchymal stem cells or fibroblasts into induced NP cells. And then we assayed them for characteristics of nucleus pulposus cells and shown that the morphology is changing with vacuolated INP cells, which is another feature of notochordal phenotype. And we have shown that they possess differential markers that are highly in the level of nucleus pulposus cells within the young human population. And we were finally narrowing down them to 3 different transcription factor combinations. And this would lead to chondrogenic and plus a nucleus pulposus phenotype. This is covered by patent now, so it is now open for discussion, and we are ready for publication in that sense. However, these populations are still heterogeneous for production of these cellular products. So in the future, we are working on to display if this type of technology can depend on NP cell product development using iPS cell technology. With that, I'd like to thank my collaborators, and happy to answer any of the questions you may have. Thank you for the opportunity today.
So thank you very much, Dr. Sakai. So we are going to address the questions at the end of the session. We've got a discussion time. So we are going to go to, so before we go to Ida, who's the next presenter, so just two things. One, please vote after the trainees presentations. So yeah, so that's one thing. And yeah, so you see that the vote will come with the name so that you don't confuse who to vote for. So the next presenter is Ida Derish. The title of the talk is Characterization of Patient-Specific Differences in Induced Pluripotent Stem Cell Derived Cardiomyocytes following hypoxia-induced injury. So Ida.
Yes, hi everyone. Thank you so much for having me here today. Is the video going to be playing, or should I present live?
You can present live.
Okay. All right. So I'm just gonna, I guess, share my slides. Okay. All right. So thank you again for having me here today. So I'm going to jump right into my presentation. So as I've repeated multiple times during seminar series, and you probably already know this, there is no curative treatment for a failing heart. And the kind of crux of this problem is that in the development of cardiovascular therapies is that current models, whether it's immortalized cell lines or animal models, really have limited clinical applicability. So our lab's goal is to kind of overcome this challenge of personalizing therapies for heart failure patients through a perhaps more reliable model, in our case, through the use of iPSCs or induced pluripotent stem cells. Now, jumping right into it, our project is part of the Heart in a Dish initiative. And so far, we've collected 143 unique samples with a ratio of healthy donors of around 1 to 10. To diseased patients. And we've also differentiated 15 iPSC lines, which we can then transform into iPSC-CMs. And it's important to note that our 15 lines have a ratio of 60% males, 40% females. They're 80% from dilated cardiomyopathy patients to 20% healthy controls. And they come from a diverse patient population, which is recruited from the heart failure clinic at the MUHC. Now, jumping into the methods, I'll go over this very quickly because we only have 5 minutes, but basically we generate iPSCs by collecting patient blood and isolating peripheral blood mononuclear cells, or PBMCs, which are then reprogrammed either through Sendai virus transfection or episomal vector transfection. And then we differentiate them into cardiomyocytes. Now, the first step is to characterize the iPSCs to make sure that they're of high quality. And we do this through 3 main methods. So the first 2 being immunostaining and RT-PCR to see whether our generated iPSCs express crucial pluripotency stemness markers, essentially. And we also started performing, thanks to Dr. Hebert's lab, performing karyotyping to check the chromosomes for any abnormalities, because again, the reprogramming step does have a tendency to have those arise. Now, once those checks are done, we differentiate the cells into iPSC cardiomyocytes, and we also perform a check to ensure that these cells express cardiac markers via immunofluorescence and RT-PCR once again. And then we move on to checking contractility as well as calcium flux, which you see in both of these videos. And both of these are important characteristics of iPSC-CMs that can be impacted after an injury in a particularly patient-specific manner. So here I'm showing to you the data from 5 lines at the baseline, meaning that we have not induced any kind of injury yet. And we took a minimum of 5 videos every day and averaged the number of beats per minute. And we can see that the iPSCCMs derived from cardiomyopathic patients seem to beat more irregularly than the controls, which I show here in green. Now, after performing these measurements, we induce hypoxia in a hypoxic chamber, of course, via an anaerobic packet, bringing down oxygen levels to around 0% for 24 hours. And what we show, and what we saw, which is what I'm showing here, is that overall we can see that the controls generally fare better in hypoxic conditions when compared to disease patients, but of course, a lot more research has to go into this. We've also looked at protein expression, so the proteins expressed by different cell lines. And as preliminary data, we can see that not only are some cardiac markers differentially expressed, but also some factors involved in heart failure might be upregulated, as we expected in cardiomyopathic patient-derived cell lines. So now we just have to figure out why that is the case. And finally, I get to kind of the data that is pointing towards, the future of this research as well. So we took 1 healthy and 1 diseased patient and live stained them with fluorophore for calcium visualization. And we took measurements every 500 microseconds under the confocal microscope. And what you can see here is the calcium flux within a culture. And in the uninjured control, we can see the calcium flux is very consistent. Whereas when we, obviously induce an injury, the frequency of the pulses, their amplitude is dysregulated. Now, looking onto the diseased patient, we can really see that there's not much rhythm, and the cells seem to be very, very disjointed. And some cells seem to kind of retain their ability to handle calcium, as shown in the graph right here, but on average, not so much. So what does this kind of mean before I get to my conclusion? Well, actually, there's not much that we can draw from this just yet, because even though the same number of cells were plated in the dish, a lot more cells die in the cardiomyopathic patient cell lines for some reason. So that's kind of a main limitation that I'm trying to get over through my research. So just in summary, I want to kind of highlight 4 main points, notably that at the baseline, generated iPSCCMs display different beating patterns and contractility. With hypoxia, iPSCCMs may display differences in metabolic activity, protein handling, protein expression, and calcium handling. The study of patient-specific iPSCCMs provides insight between healthy controls and cardiomyopathic patients. And finally, very importantly, a larger population of iPSCCMs must be studied to draw significant conclusions. And with this, I just want to say thank you to my lab, to my collaborators, and as well to all of the funding agencies that make this research possible. Thank you so much.
Thank you very much, Ida, for that wonderful presentation. The next speaker is Lily, and the title is All Vanillin Enhanced Mesenchymal Stem Cell Differentiation and Disc Cell Phenotype. Lily?
Yep. Good morning, everyone. Thank you for having me. Low back pain is the leading cause of disability worldwide. Disc degeneration is one of the main causes of low back pain. As we see in Figure A, degenerate IVDs lost water content, proteoglycans, and disc height, which may cause the pain. Human mesenchymal stem cell therapy has been intensively studied in the field to improve disc health. Figure B shows the interaction between MSCs and disc cells. MSCs produce trophic factors to improve disc cell phenotype, and the disc cells induce MSCs differentiate towards disc-like cells. Our group previously found that the anti-inflammatory natural compound vanillin reduced the senescent cells in IVDs. Therefore, we further explore in this study whether vanillin could enhance the interplay between MSCs and disc cells and improve stem cell therapy for IVD repair. We firstly verified the vesicle transfer between MSCs and disc cells. We collected the conditioned media from red stained cells with and without vanillin treatment and applied them to the other type of non-stained cells for 7 days. We found that vanillin increased MSC release vesicles and increased disc cell uptake vesicles, shown in Figure A. And also vanillin also increased the vesicle release from disc cells and the vesicle uptake of MSCs, shown in figure B. The fluorescence quantification in figure C and D validated also vanillin increased vesicle transfer from MSCs to disc cells and vice versa. We evaluated the gene expression of MSCs and disc cells in 3D pellet cultures after transferring their conditioned media. We validated that MSCs differentiate towards disc-like cells when exposed to the conditioned media of disc cells, shown in figure A. We found that also vanillin further promoted MSC differentiation, shown in figure B. In addition, we validated that conditioned media from MSCs improved disc cell phenotype, shown in figure C. And we found that also vanillin further promoted disc cell phenotype improvement, shown in figure D. To have closer cell-cell contact, we performed the co-culture in the following experiments. We then evaluated the inflammatory condition by ELISA and found that also vanillin suppressed the inflammatory cytokine interleukin 6 and 8 production from both MSCs and disc cells. We detected sulfated glycosaminoglycan production in conditioned media and proteoglycan deposition on cell pellets, as shown in figure A. With 50% disc cells, the 1-to-1 co-culture pellets produced more SGAG, indicating MSC promoted SGAG production, and this effect was enhanced by also vanillin treatment. The red intensity in figure B represents proteoglycan deposition on pellets, which was increased by MSCs as well. This effect was also enhanced by also vanillin treatment. Senescent cells are associated with disc degeneration, as the accumulations of senescent cells contribute to the inflammatory environment. So we evaluated cell senescence on pellets by P16 staining. The quantification of P16 positive cells showed a significant decrease of senescent cells in 1-to-1 co-culture pellets compared to that of the mean of MSC and the disc cell pellets, indicating co-culture with MSCs reduced the senescent cells. In addition, figure B showed that also vanillin further reduced the senescent cells on top of the co-culture effect. Therefore, we can conclude that disc cell conditioned media induced MSC to differentiate towards disc-like cells. Conditioned media of MSCs improved disc cell phenotype. MSCs promoted proteoglycan production and deposition in co-cultures. And all this were enhanced by also vanillin. In addition, also vanillin increased the vesicle communication and suppressed the inflammatory cytokine production in both MSCs and disc cells. I would like to thank my supervisor, Dr. Hageland, my colleagues, and all the funding agencies for their support. Thank you.
Well, thank you very much, Lily. So is Kusum here? So if Kusum is not here, I think we're gonna play the recording. VP Solutions, please.
Hi, I'm Kusum, and today I'll be presenting a project which is on dissecting the stromal drivers of gastroesophageal adenocarcinoma chemoresistance, also known as GEA. So the standard of care treatment for GEA is docetaxel-based triplet chemotherapy, also known as DCF. And the main problem that 50% of patients experience is recurrence of their cancer due to chemo resistance. This has led researchers to shift their focus to the tumor microenvironment to identify drivers of chemo resistance. Cancer-associated fibroblasts, or CAFs, are the most abundant cell type in the tumor microenvironment. Fibroblasts are normally found in their quiescent state and become activated upon the site of injury, causing them to express different markers and change their morphology, as we see on the right. Once the wound has healed, the fibroblasts can undergo apoptosis or revert back to their dormant state. However, in cancer, this process becomes chronically activated and can promote tumor progression. As seen in other cancers, we hypothesize that heterogeneous CAF populations may be responsible for patients' response to chemotherapy in GEA. In order to study this, I'll be looking at the following objectives. First, I'll be selecting my patient cohort with their matched treatment naive and post-chemotherapy tumor organoids and fibroblasts. I'll then be using an already available single-cell browser containing data from over 30 GEA patients to identify CAF markers, which will then be used to experimentally characterize the patient's CAF populations and correlate that to their tumor response. Finally, I'll be performing a heterotypic recombinant co-culture between GA patient-derived organoids, or PDOs, from a good responder patient with the fibroblast from a poor responder, and then treating that with DCF to determine the fibroblast's effect on PDO's response. So here we have the single cell browser where each cluster identifies a different cell population based on gene expression, and the colors identify the cell type. For my project, I focused on the fibroblast cluster, and as you can see, each dot represents an individual patient's fibroblast. We were able to see the differential gene expression of a select 7 patients, as we see on this heat map on the right. So we have these 2 good responders, 4 poor responders, and 1 treatment-naive patient. And this heat map is based on a Z-score, where a high Z-score would indicate that a gene is being differentially expressed, either by being increased or decreased. And a low or undefined Z-score indicates that the patient either does not express this gene or that there is no change in the gene expression. So just to focus on one gene, here we have vimentin, which is a structural protein normally found in fibroblasts. From our data, we can see that the good responders have an undefined Z-score, meaning that the gene may be expressed at the basal level. While in poor responders, this gene is being differentially expressed, as seen through the high Z-score. Several studies have actually shown that vimentin overexpression is associated with poor overall survival, and we suspect that our poor responders are overexpressing this gene. And from this data overall, we can see that good and poor responders have certain genes that they are differentially expressing, and this can then be used as markers to identify distinct CAF populations. Next, I wanted to correlate the patient's fibroblasts to their tumor response. So here in blue, we have all of the patient's fibroblasts, and if you look closely, there are cells highlighted in black, which are the fibroblasts coming from poor responders listed on the right over here. And these poor responders samples are from their pre-treatment. When compared to the poor responders post-treatment fibroblasts, we actually see that there's a significant increase in CAFs following treatment, and new populations have actually shown up. This indicates that there is a change in the tumor microenvironment following chemotherapy, and it is possible that these fibroblasts are involved in modulating the resistance that these poor responders are experiencing. Here on the bottom, we see a comparison of the different cell types between pre and post-treatment poor responder samples. We actually see that there's a more diverse population of cells in the pre-treatment sample, but in post-treatment, the majority of cells are fibroblasts, and there is a lack of other cell types. Currently, I'm now working on setting up my co-culture to see the effect of fibroblasts on the tumor organoids. And so here we have a co-culture between a good responder organoid with poor responder fibroblasts. The domes, as you see, are the tumor organoids, and these web-like structures are the fibroblasts. And you can see that they're actually interacting the way that the fibroblasts are attaching to the periphery of the organoids. We then treated this co-culture with 1,000 nanomoles of DCF, and we see that the organoids have actually shrunk in size, and some have even exploded, indicating that they are dying as a result of the chemotherapy. What we want to see now is whether these fibroblasts from the poor responder patient are affecting or changing the chemo response of a good responder organoid to a poor response. And so to conclude, good responder patients, as seen from our data, express distinct CAF markers from poor responder patients, and poor responder patients demonstrate a greater expression of CAFs following chemotherapy. So this is the end of my presentation, and I'd like to thank everyone listed on the screen for helping me with my project so far. Thank you.
That's great. So the last speaker in this session before the discussion is Claudia, and the title is Increased Urinary Ratio BDNF, pro-BDNF in a female population with overactive bladder syndrome. Claudia.
Thank you. Good morning, everyone, and thank you for having me here today. Well, to begin with, there we go. To begin with, what is overactive bladder syndrome? Well, it is a clinical entity that is characterized by urgency, increased urinary frequency, with or without incontinence. It is associated with aging, affecting both male and females, and it also carries a significant loss of quality of life. Now, there have been several studies looking at the molecular basis of bladder function, and among the most studied elements are the family of neurotrophins. For this particular study, we will focus on BDNF in both its precursor and mature forms, and also their respective receptors. Now, in this schematic, I want to highlight the conversion of pro to mature form that is regulated by plasmin and several metalloproteases. We can see that pro-BDNF has an affinity to the apoptotic receptor P75, and that the mature form has a higher affinity to the receptor TRKB, which is associated with neuroregeneration. Additionally, some studies have reported several microRNAs that are implicated in both direct and indirect upstream signaling of the BDNF mRNA. So given the gap of knowledge in this area, we aim to compare the amounts of specific neurotrophins along with the MMPs associated with its metabolism in order to establish a comparative correlation between OAB and healthy controls. For patient recruitment, it was done at the urology department of the Jewish General Hospital. Our sample size was a total of 40 female participants between the ages of 50 and 80 years of age, subdivided into each group. We excluded cases associated with malignancy, neurodegenerative disorders known to cause neurogenic bladder, such as multiple sclerosis, amongst other secondary causes of OAB. Now, we used validated clinical questionnaires to measure urinary frequency, the severity, and also presence or absence of urge incontinence. Next, we collected 2 early morning urine samples, one to rule out the presence of concurrent UTI, which can mimic the symptoms of OAB, and the second sample designated for exploratory biomarker study. Additionally, we collected an overnight fasting blood sample, specifically looking to find parameters associated with metabolic syndrome and renal function status. Our measurements were conducted using ELISA technique, PCR, and specific enzymatic kits. Now, for our results, this table summarizes the clinical characteristics of our cohort. Compared to the control group, the OAB group was found to have a significantly higher HOMA for insulin resistance and decreased eGFR, which reflected impaired renal function. The overall presence of metabolic syndrome and hypertension was also found to be significantly higher in the OAB group. Now, continuing with our results, looking at this from left to right, we can see that BDNF levels were not different amongst the groups. In the middle, we can see that pro-BDNF was statistically lower in the OAB group, but most importantly, the ratio was found to be much higher in the OAB group, as seen on figure C. We then conducted ROC curves used to evaluate the diagnostic value for OAB. And these findings suggest that the ratio of mature to pro form has the highest predictive value for diagnosis of OAB. As we can see, it's approximating to 0.8. Now, for the microRNA levels, these were not statistically significant amongst both groups. However, as MMP9 is one of the major enzymes that converts pro to mature form of BDNF, we measured its activity and found an increase in the OAB group. Additionally, there's a specific gene called the MIR491-5P, which inhibits the translation of MMP9 mRNA. And we can see that it was decreased in the OAB group, which confirmed the observed activity in MMP9. So to conclude, these preliminary results indicate that the ratio of mature to pro BDNF in urine might actually be a better indicator of OAB than the neurotrophin alone. And this was further matched by the increase in MMP9 and somatic activity as stated in the previous slide. This study essentially is a preamble for furthering a better understanding of the etiology and the severity of OAB, as well as differentiating what is pathological and what is physiological in the aging process. So I think for future studies should emphasize on larger sample sizes, include both genders, amongst other factors that were not included in this cohort. Lastly, I would like to acknowledge my supervisor, Dr. Lisan Campo, and the lab members, as well as all the funding agencies that made this research possible. Thank you very much for your attention.
So thank you very much, Claudia. So we are quite very ahead of time, and we have 25 minutes for discussion. So feel free, please, to raise your hands or just unmute and ask any of our speakers. Any questions? So Dr. Sakai, can you comment on what the stem cells are doing? Is it the stem cell differentiation, or is it the factors that are coming from the stem cells?
You mean cell transplantation? In terms of cell transplantation, they can have multiple potential effects. cells can survive and be functional as NP cells. So they can, stimulate the native cells to be more active. They can secrete anti-inflammatory factors, try to normalize the degenerative process in various ways. And this has been shown by different in vivo data, showing that after cell transplantation, the degenerative process may be reversed to some extent.
Okay. And how long does that effect stay? The effect of the stem cell? Yeah.
So if we use mesenchymal stem cells, the cells that can survive and be active for quite a certain number of duration is limited. In our experience, it's around 15 to 20% of the cells among all the cells that were transplanted in the mesenchymal stem cells were active for around 2-3 weeks. However, if we injected progenitor cells of the nucleus pulposus progenitor cells, they can last for around 3 months or greater than that as well. So it's all about the number of cells that can survive and be accommodated to the specialized niche of the intervertebral disc.
Thank you. Dr. Bartlett?
Yeah. Again, sorry, it's a question for Dr. Sakai, but it was eating away at me while you were talking. Has anyone ever taken transplanted a nucleus from a very young, presumably animal, to an old animal, and then demonstrated a restoration of regenerative ability to any great extent?
Thank you. Thanks for your question. There is not an animal study directly comparing young cells versus old cells, but we have injected young human cells versus old human cells into immunosuppressed or compromised animal models, which shows that the older cells have limited capacity to regenerate the disc itself. Thank you.
Okay, thank you.
Any other questions?
Yeah, I could thank you, Dr. Sakai, for a very interesting talk. I was just wondering, could you comment on the secretome of the BMSCs versus the cells themselves?
So thank you for your question. To answer your question directly, we have not looked into like secretomes or exosomes from the NP progenitor cells that we have identified and isolated, which we need to maybe define that in the future research question. There's lots of MSC-related exosomes, which are influenced by interaction with intervertebral disc cells. And there's lots of microRNAs that are involved in that process as well, which are in the literature. So I would hope and think that there is a lot of exchange of these factors ongoing with NP progenitors itself, but I cannot answer you directly at the moment. Thank you.
Thank you.
So can you use the Tie2 positive stem cells for cartilage repair?
I think we have looked into articular cartilage for Tie2 positive cells. There are Tie2 positive cells, very scarce percentage, around maybe less than 10% within the young cartilage as well. But we don't know the exact function and what it's doing in the articular cartilage homeostasis at the moment. That's another future research question as well. Thank you.
Any other questions?
Yeah, hi, Dr. Sky. Amazing as usual, and I'm always impressed with the clinical development. I think it's amazing to have this ongoing and as a potential treatment for discogenic pain. I just had a question. It's related to actually the pain part. So do you know the mechanism of how the MSCs, your MSCs inhibit pain over time?
Sure. The painful chronic disc has, repetitive, since the disc is contained structure, if once the inflammatory responses start to take part, you can see from the imaging and also from the sustained patient condition, you need something of a game changer within that inflammatory process. And MSCs can provide like exosomes or anti-inflammatory agents and cytokines that can access the catabolism towards more anti-inflammatory response. And we see that, there's proteoglycans in ECM restructure also occurring in that process as well. But as said before, MSCs have the limited capacity to structurally regenerate the tissue itself. Maybe directly, you may be trying to wanting to know about the structural mechanism of certain pathways, but certainly NF-kappa B pathway, other major inflammatory pathways like the TNF, stimulating the NF-kappa B downstream. These type of inflammatory responses are augmented by MSC transplantation as well.
Yeah, it's quite amazing. I had a question for a student, one of the students, Lee, on the Ovanillin treatments. Yeah, it's very nice. Very nice work. So I was wondering, so you, Ovanillin you showed that it was able to inhibit IL-6 and IL-1, I believe.
IL-8.
And
IL-6 and IL-8.
Oh, IL-8, sorry. So I was wondering if you inhibited these cytokines, can you replicate to some extent the properties of Ovanillin to try to tease out its mechanism?
Sorry. Do you mean if we explore how Ovanillin inhibited interleukin 6 and 8?
Well, like if does inhibition of these cytokines alone, can that replicate Ovanillin effects, or is it really other properties that are leading to the effect of Ovanillin on disc or MSC differentiation into NP-like cells?
So in this particular study, we didn't look into the other pathways, but one of our previous studies showed that also vanillin can inhibit toll-like receptor 2. So I would think that's part of the role of how also vanillin modulate inflammatory cytokines.
It's through the inhibition of toll-like receptors.
Yeah.
But they have I guess because they have downstream signaling pathways. I was curious on the similarities and whether these signaling pathways may be related. And if you just inhibit these pathways, can you just replicate some of the effects of Ovanillin?
We need to further investigate into those directions.
No, it's very interesting though. But thanks.
Any other questions for we have other students. So Ida, so if you get your IPS, one of the things now is trying to find if there are differences between male and female. And have you looked at your IPS? Are there any differences, or from different patients with age, for example? Because I think you look at it, patient-specific differences.
Yeah, so right now, because we have such a small population, I don't want to make generalizations that would be, false in a larger population, which is why I didn't really say, female patients have a tendency to have this, male patients have a tendency to do that, because right now, to be honest, we don't have enough power, like we don't have enough patients to really do that. I think by the end of my PhD, I definitely hope to have some kind of conclusions based specifically on biological sex, maybe on age as well, on different genetic backgrounds. So in our kind of patient population that we've recruited at the heart failure clinic, we're actually very lucky to have patients coming from various different backgrounds, and so I really do hope eventually I'll be able to say that. But I think my first priority will definitely be to essentially separate the biological sexes, because we know that there are differences in male patients and female patients with relation to treatment. So it would also be interesting to see how different they are in terms of injury and recovery, of course. But yeah, so it's kind of an ongoing work. What I presented today was just kind of a preliminary, exciting outlook on just a couple of patients so far. Yeah, did I answer your question?
Yes, that's great. Thank you very much. Dr. Barrett?
Dr. Wiley. Yeah, I don't. It's not really, none of these things are really my area, so I can't really think of any, nothing sprung to mind.
Okay, so that's an odd hand.
There's a question from Nicoletta, yeah.
Nicoletta, yeah.
Yes, thank you. Dr. Sakai, thank you so much for your wonderful presentation. With regards to the mesenchymal stem cells, have you investigated MSCs from different tissue sources? Would you expect that those from the bone marrow would have the highest effect? I'd like to have your feedback on that, your suggestions. Thank you.
Thank you for your question. Actually, we've only investigated bone marrow derived MSCs as ourselves, but other researchers have looked into adipose derived MSCs and other sources as well. It doesn't really seem to have a large difference among the source of MSCs due to the limited effect of MSC itself. So, in order to answer your question, I do not have data myself comparing directly between different sources for MSCs. Thank you.
Okay, thank you. And another follow-up question, do you foresee a time when instead of the cell therapy that the derived factors from these cells would be used clinically?
Thank you. As you know, PRP is already used on other exosomes, and other factors secreted by MSCs are also tested in vivo animal models. So I would forecast that there will be some vendors or people who will be trying to commercialize in that respect. Thank you.
Thank you.
So Claudia, why is this ratio important why not other neurotrophins, the ratio between BDNF and pro-BDNF, which is the zymogen?
Right. So first of all, OAB is a clinical entity, and we don't currently have anything objectively to diagnose it. So we rely heavily on what the patient tells us. And although we do have validated questionnaires to sort of like place them into a severity category, or if they have or don't have OAB. This is highly subjective in the clinic, and it can change. There's a lot of confounding factors that can change the answers and the way these patients answer these questionnaires. So we want to objectivize the way we diagnose, the way we follow up, and also the way we can see how treatment is working or not. So the neurotrophins, since they've already been categorized as potential biomarkers, it's important to see, especially the relationship from what I kind of concluded, if it's pathological versus physiological changes in aging. And I think that's why they're very important, especially in the aging population, because we don't currently have any objective tool.
Okay, so what about the ratio of NGF?
Right, so there's other neurotrophins. We decided to focus on BDNF because it's the most abundant in the human body. So there's been studies that show NGF, which is another neurotrophin, a very important one. And actually our lab did some studies on NGF, and they also found that the ratio was more predictive, or that the ROC curves were more predictive than just the neurotrophin alone. So the ratio is important because it kind of gives us an idea of how we can target therapeutically these ratios to bring back a right balance to help alleviate with symptoms. Now, obviously, these treatments have not been tested in humans yet, but our laboratory is working on animal models to see if we can target these pathways of the metalloproteases and the genes involved in their regulation, so that we can restore the neurotrophin ratios.
Okay, thanks. Dr. Philip?
Yeah, I have a question for Coulson. She's there? Yeah, she's there now. Okay, I enjoyed your presentation. You told us that calves are activated fibroblasts. And I was wondering whether you could tell us about any differences between calves versus the other major fibrotic phenotype of fibroblasts, which is in organ fibrosis, like in keloids or any organ, lung fibrosis, other fibrotic states.
So the main difference, like in terms of phenotype, it really is the fact that in cancer it's really chronically activated. I'm not sure in terms of like organ fibrosis how it would be, but the activation process is expected to be similar. It's just that in cancer it would be more chronic and it would be long-term.
Okay, so the resolution does not happen. So these are sort of probably at the level of signaling pathways.
Mm-hmm. And also like just interactions with the tumor can probably further exacerbate that activation. So
yeah, very good. Thank you.
Okay.
Any other questions? We still have time. Nicoletta?
Yes, thank you. I have a question for Lee.
Yes.
Yes, I enjoyed your presentation. Perhaps I missed this part. When you spoke about the O-vanillin enhancement of the MSCs, how was this done? Can you, perhaps I missed it. You expose it for how long to the MSCs or?
Yes, for the gene expression part. So we cultured the cells individually for 28 days in total. So the also within treatment is at the first 4 days. Then the rest of the days will be the regular culture with the regular complete culture media. So that then during each 4 days we transfer the conditioned media from disc cells to stem cells, and also from stem cells to disc cells. After this period of time, we started to evaluate the gene expression change.
Okay, thank you. So the exposure to vanillin was about 4 days, and then they were back in regular culture media. Okay,
Yes,
thank you
exactly.
very much.
Thank you.
Any other questions? So I think if there are no other questions, I would like to thank all the speakers for wonderful presentations. Thank you very much, Dr. Sakai. I know that it's you're about to sleep over there. Thank you very much for everyone for attending. And so there is a break, which goes up to 10:30. So we have 10 minutes break, and then we'll start the session 2 at 10:30 with Dr. Giulio Fiore. So thank you very much, everyone.
I hope you guys can hear me well. I think so. So yeah, I really appreciate the opportunity to chair this session. And as requested by the organizers, the structure of this session will be a bit different compared to the previous one. We'll have two distinct discussion periods, one right after the keynote presentation and another one after the student presentations. So I hope that's okay for everyone. So to start, it's a real pleasure to introduce our second keynote speaker, Dr. Jason Harley. Dr. Harley is a tenure-track assistant professor at McGill's Department of Surgery, where they lead an outstanding research program aimed to enhance surgical and health professional education, and also support healthcare workers in reducing adverse events and inefficiencies associated with unregulated emotions, burnout, and harassment. Dr. Harley completed a PhD in educational psychology at McGill, followed by a postdoctoral fellowship in computer sciences at the University of Montreal. And they were a tenure-track assistant professor at the University of Alberta before returning to McGill in 2019 to fund the sales lab. And in the lab, Dr. Harley uses qualitative and quantitative methods to measure cognition and emotions associated with a variety of surgical and medical competencies. And their work leverages a wide range of technologies, including virtual reality and artificial intelligence, to support the education and skills acquisition of healthcare workers. And Dr. Harley received numerous prestigious awards and research grants throughout their career, and their research and teaching have led to appearances in numerous major media outlets in Canada and also internationally. So it's a real pleasure to have Dr. Harley here today to present this keynote lecture focused on the role of simulation and emerging technology in supporting surgical training and psychological well-being. Dr. Harley, the floor is yours.
Thanks so much for the introduction, Julio. And I think the video is going to play. Hi everyone, I'm Jason Harley, an assistant professor in the Department of Surgery and Sciences at the Research Institute of the McGill University Health Center. I'm also the director of the Simulation, Affect, Innovation, Learning, and Surgery Lab, director of research at the Steinberg Center for Simulation and Interactive Learning, and an associate member of the Institute for Health Science Education. And I am pleased to talk to you today about the role of simulation and emerging technology in supporting surgical training and psychological well-being. Thanks so much for having me as a speaker at Experimental Surgery Research Day. So first, I'll provide a brief disclosure. I'll give a little bit of an overview in terms of psychological wellness, or the lack thereof, as some might say. I'm probably one of those people. Before talking about ways that we can take action through research, and also what's next. So a disclosure that Oxford Medical Simulation, whose software I will discuss, are a research partner on one of my grants and are supporting the corresponding research project through in-kind contributions. So we know that there are pre-existing challenges to healthcare professionals' psychological well-being. These range from demands and shortcomings of the healthcare system and resulted in healthcare professionals' absences, suicides, and medical errors. The ability to cope has been further compromised by stresses and sources of stress from COVID-19, and these stresses have highlighted a lack of mental health support for healthcare professionals. So again, things were bad before, and they've been more challenging with COVID. We know that a lack of early access to therapy and a restriction of adaptive coping mechanisms to assist in managing stress can result in psychological distress. So we're talking about distress, we're talking about things like anxiety, depression, burnout during a pandemic, and carry long-standing adverse consequences So this is something we want and need to do something about. So following the SARS outbreak, many healthcare professionals continued to experience related stress reactions, similar to post-traumatic stress and depression. Healthcare professionals' well-being has been threatened by COVID-19 in important ways, and there's been lots of challenges as a result. So there's been an urgent need to create and adapt to new protocols, make difficult decisions. People have been concerned about their own personal safety, fears for their own and family's health, and unfortunately, the death of loved ones, friends, and colleagues. From some of the research, we've seen that a considerable proportion of healthcare professionals reported symptoms of depression, anxiety, and distress, especially those in Wuhan and frontline healthcare professionals who are directly engaged with patients with suspected or confirmed COVID-19. Others identified high rates of stress-related symptoms, including depression and anxiety, among a large study of over 1,500 healthcare professionals in China. So research in Italy identified that 86% of healthcare professionals surveyed, nearly 1,000, experienced moderate to severe levels of psychological distress during COVID. And again, this was some of the early research that came out, and later research has confirmed these findings. So again, we've also seen not just in journal articles, but also in mainstream news that things have been difficult. Myself and my research team have been part of helping to highlight the challenges to psychological well-being that healthcare professionals, both physicians and nurses, have been facing during the pandemic. This is just a subset of some of the media that we've done based on a study we did that was funded by MI4. We've published 2 empirical articles from that MI4 study that I'll talk briefly about. And we have a really great research team with some familiar faces, such as Dr. Leanne Feldman and Dr. Gerald Fried, as well as colleagues from psychology, counseling, nursing, etc. Some of the things we found by conducting a survey here at the McGill University Health Center was that there was high stress and psychological distress incidence rates before COVID-19. So we asked people how they were feeling pre-COVID-19. But although stress and psychological distress was high before COVID, it was even higher, statistically significantly so, with both stress and psychological distress incidence rates during the pandemic. So we asked people how they felt before, to remember how they felt before the pandemic or before COVID, and then how they felt at the moment. And that was again during COVID-19. We also found that there were consequences on a variety of levels stemming from these psychological stresses that people were experiencing. Physicians and nurses reported that stress negatively impacted their perceived work performance, including self-doubt and patient care. And among the respondents, half of the nurses and 1 in 5 of the physicians expressed an intention to quit their job. So again, some clear indications that there are consequences in addition to the psychological discomfort that burnout, stress, depression, and anxiety can leave us with. One of the interesting findings we had from this study was that support being available, such as counseling services, did not mean that support was necessarily used. So within our sample of healthcare workers, we noted previously that there was a high level of psychological distress across various forms, as well as high level of stress reported during the pandemic. We asked people a number of questions. We asked them, first of all, whether they were aware of counseling services, and many indicated that they were. Not all of them, but many did. Then we also asked people kind of what they thought about these services, whether they were helpful or not, and many people indicated that they were. But when asked whether they were using these services, we found a gap there. So again, just because someone is aware of something and perhaps is aware that these sources of support can make a difference, doesn't mean that people are going to elect to use them themselves. So this is something that is important not just for general healthcare workers and healthcare psychologists, etc., to be aware of. But also for leadership within hospitals and other settings to make that they need a little bit more information sometimes to make nuanced decisions. We found also that healthcare professionals who use more adaptive coping strategies or buffered against the effect of stress on burnout and turnover intentions. So again, this highlighted that adaptive coping strategies are indeed, are effective and can help with some of the consequences that are associated with some of these negative psychological experiences. So we did find, as I mentioned, that adaptive coping strategies helped, but we do need to do more than download mental health responsibilities to healthcare workers and trainees who are, as we saw, already quite stressed and, as we know, also very busy. So we know that the availability of hospital-level support toward COVID was rated as the most useful resource to buffer against the stress effect on burnout, psychological distress, and turnover intentions. So again, hospital-level support, it does make a difference. We want to avoid downloading these things onto healthcare workers and trainees. So knowing this, what are some of the things that we can do? Well, to take action, one of the things we can do is move the needle earlier a little bit. And that can, and formal education can play a role, particularly simulation. So some of the things we can consider are what are effective and ineffective coping strategies in healthcare contexts. So this is one of the ingredients in order for us to integrate some of these psychological well-being best practices into formal education. The good news here is that we have the literature that supports and makes clear to us what kinds of coping strategies are adaptive and which ones are not adaptive and are going to lead to the kinds of outcomes that we want to not be supporting and providing. We also have the psychological models available. There's also a need for practice selecting and using these sorts of coping strategies in high fidelity simulations. We have the high fidelity simulations. One of the things we know about them is that simulations provide an authentic environment, meaning that the emotions aroused are typically more intense, and this provides us with a great option for giving trainees or people who are engaging in continuing professional development an opportunity to practice, dealing with stressful situations, stressful things, new procedures in a safe and standardized context. What we don't have is really anything explicit or formal in terms of making people aware or giving people feedback or encouraging people to reflect on the types of coping strategies that they're using. This is something I'll talk a little bit about later as one of the research projects we're engaging in with my lab. Similarly, high quality feedback on the practice coping with emotions. So we have some of the ingredients. So we've got lots of ingredients for one, and we've got some of the ingredients for two, and I would argue that, again, 2 or 3 are things we need to move the needle forward on. So I'll briefly talk about the models. One of those models that is, I think, a crucial ingredient for us better understanding how to support healthcare workers specifically is a model that my colleagues and I recently published that was situated specifically in the context of the pandemic. And we adapted an existing model that had been published back around 2009, 2010, more that was an industrial psychology model, and we extended it quite a bit to account for some of the specificities of healthcare workers, such as dealing with a variety of different types of outcomes, the existence of support, context, etc. I won't get into that. I invite you to read the article if you're interested in the details. What I really want to draw your attention to here is the place where coping strategies can particularly make a difference is in helping to mitigate the effect of stress on the psychological distress that people experience. So in other words, if healthcare professionals and healthcare trainees have practice selecting and using good adaptive coping strategies, such as rethinking how they're framing a situation, as opposed to relying on getting a drink the moment they're out of an OR or a bad session. This can make a difference in better responding to stress, which is something that's a really critical skill, not just for one's own well-being, but as the model shows, for patient outcomes, for the more long-term well-being of healthcare professionals, and also for to help mitigate the adverse institutional outcomes that come, for example, from people quitting or not being available to come into work. We also note too in this model the importance that context plays, both at an individual level and at a more organizational macro level. Again, I won't go into too much detail with this model during this talk, but I'm happy to take questions about it. And this actually model was featured this week in the McGill University Health Center's news and newsletter. We're talking about emotion regulation. Emotion regulation strategies and coping strategies do have some overlap in terms of what they are. This is another model that I published in 2019 with colleagues, and we were interested here to situate emotion regulation strategies and how we think about emotion regulation as a psychological process, and bring this into the context of education and training, rather than the kind of limited low ecological validity context that characterize a lot of experimental psychology studies, where we don't really have a lot of details in terms of what are the emotional dimensions of an environment and what are the affordances and constraints that trainees and others have. In other words, are all emotion regulation strategies appropriate? And certainly when we think about emotion regulation strategies that are and are not available to us in a healthcare context, whether it's a simulation or an operating room, there are a number of things that are not so much there. There's, if one is in the operating room and something happens that's distressing, electing to leave the operating room, if we're part of the operating procedure, could have really dire consequences, to the patient and to the ability of the team to support that patient outcome. So again, that's not really something that's on the table as much as it would be in a typical scenario, as just one example. Indeed, the emotion regulation strategies that are more available to us are those that are internal. What can we do to focus our attention back on the task, say, change how we're thinking about the particular situation, perhaps decatastrophizing it, reminding ourselves what kind of control we have, and perhaps also, depending on what our role is in that particular situation, looking for constructive ways to modify it. So these are just some examples. Again, this isn't a theoretical talk. I'm happy just to answer more questions about this or other models at the end. There's also a number of methods that we have available to us to implement some of these directions with the purpose of trying to support, again, psychological well-being in surgical education. So some of those things are new assessment technologies and techniques. These technologies can provide us with a way of visualizing what emotions look like while people are engaged in simulations or while they're engaged in training or authentic clinical cases. Physiological sensors provide us with an index of emotional arousal, and this can give us a sense of whether people, for example, are having spikes of anxiety or spikes of frustration in response to something that's happened, which can, depending on the experimental protocol, depending on the software, etc., which can be visualized in real time for people, including for simulation educators, to give them feedback in terms of what's going on. And in the case of a simulation, if something looks like it's starting to spiral out of control, perhaps again elect to pause things, or at least make a note to remember that moment and make sure that in the debriefing it comes back and is something that's talked about and debriefed on. Audio and semantic analysis can give us information in terms of what was going on. These are things, this can be verbal cues that people, so what kind of words people are using, how are they valenced, and what kinds of speech characteristics are people using. And there's interesting applications for machine learning and other approaches that we can use, in addition to, of course, human coding for this. Machine learning can also be used to look at posture, which we can get from video data. This again depends on, what is the setting like. People's posture might be a little bit more standardized during an OR or simulation procedure. During a debrief, that could be a little bit more telling. We can also use a combination of AI techniques such as artificial neural networks with facial recognition software when we're not wearing masks to get a sense of what basic emotions individuals are displaying. This provides us with just another metric for getting this kind of information. Finally, eye tracking, which can also be taken from video, although typically it's taken from videos where the camera is directed towards individuals' eyes so that we can see what people are seeing. This gives us a sense of where their attention is. And where their attention is, this is typically, research has shown where people's thoughts are, unless mind wandering is happening, and this can help us to make help us narrow our inferences in terms of why people are having an emotional reaction. So this is really rich information for us to bring together, along with self-reports, along with interviews. What's going on and how we can better support people, but also in an educational situation, how we can bring these channels in for feedback. And indeed, this is something that we're doing with a particular study, and one of those channels is the physiological. And we're particularly interested in having teams in simulation context, and we're doing this at the Steinberg Center for Simulation and Interactive Learning, amongst others, and getting a sense of how the emotional reactions of one cascades or does not with regards to others. Does it make a difference if the leader is experiencing a spike in emotional activation, such as frustration or anxiety? Is there an emotional contagion with the team members? How does that leader regulate those emotions? Does that make a difference visibly with the emotions in terms of physiological activation that others in the team are experiencing? And as I mentioned, we can use these tools in addition to self-reports to provide prompts for debriefings. One of the things we've done is developed a new self-report or survey tool that asks simulation participants questions about how they regulated their emotions, what kind of strategies they used. Was there a particular event in a simulation that was emotionally salient with regards to being challenging? And whether they thought the emotion regulation strategy that they used was effective in dealing with the emotions that aroused from it, and whether there were differences between how they approached handling the challenges experienced from this particular moment in the simulation versus how they thought their group dealt with it. These provide great prompts to get people to think about how they felt, how they reacted, and reactions being the first part of the Pearls framework. So already emotions are embedded, at least with this framework, in the debriefings, but it gives us an opportunity to probe a little bit deeper, and we think have people come to the debriefings a little bit more prepared to talk about some of the emotional dimensions and also how those emotional dimensions impacted other performance pieces. So that's something, and that's research we're doing right now, and certainly there's very little work that's been done on emotion regulation in simulation contexts. So this is quite interesting also to get a sense of how this work is capturing and characterizing the nature of emotion regulation dynamics, especially in a team setting. So some members of my team are going to, I believe, share this research later today. So there's also a variety of tools and environments that we're using this sort of that we can pursue these research questions in and provide people with opportunities to, again, regulate their emotions. So high fidelity simulations are one of them. And as I mentioned, with greater fidelity comes higher intensity emotions and therefore better opportunities to practice managing those emotions. High fidelity mannequins provide us with opportunities to do so. They provide often visualizations of vitals that can be expressed through physiology and monitors, as we see in the left-hand pane. And in these settings, there's often windows or mirrors, as well as cameras and microphones that help us to do high-quality audiovisual monitoring. And this allows us to get, again, great quality and rich data in terms of how a team is performing when they're interacting with these high-fidelity mannequins. There's also a variety of virtual ways that people can interact with patients, and this is a different type of standardized patient where they're standardized because they're driven by artificial intelligence, typically some kind of a decision tree. For those of you who play video or computer games, who are playing basically a non-playable character or NPC role. As I mentioned, we're doing research with such an environment, Oxford Medical Simulation, which provides people with an opportunity to be immersed in the environment, whether they're using a screen or a virtual reality headset, and take advantage of automatic performance scoring, as we can see on the top left-hand corner. These environments, a little bit like simulation environments or in-person ones, provide a safe learning environment, but they do have the benefit of being individual generally. So people can often focus on their own performance, even if they're interacting with a team of virtual characters. There's interesting affordances with these technologies with regards to opportunities to engage in distance or on demand, in other words, repeat learning opportunities. At the moment, this type of environment is best for procedural skills when it comes to surgery because there's not haptic feedback isn't something that's really been deeply explored or popularized in terms of the hardware side of these systems, so that kind of limits things. That being said, my lab is in the early stages of a partnership with a haptics group to investigate the role that haptics can play in surgical education training. So I hope to talk to you about that in the near future. We're also doing some research to disentangle whether screen or headset is better. This has implications for what we actually do with these environments in terms of whether we ask students to wear a VR headset or whether screens are good enough. Certainly, most people have access to a screen. Certainly not everyone has access to a VR headset. VR headsets are more expensive, so it's of course important to be able to identify advantages and ideally statistically significant advantages if we're going to recommend that people are using VR headsets. So another study that we're in the early phases of doing at the moment. We're interested too in, this research is helping to develop best practices for integrating this kind of simulation into curriculums. We're asking questions like, what is the role of the simulation instructor? Should the simulation be a standalone or should it be integrated with existing simulations? There's a lot of open questions, which makes this a really interesting and exciting avenue of research. When it comes to artificial intelligence in simulation, we think there's a lot that can be done. We recently completed a review of artificial intelligence in simulation published in the American Journal of Surgery. And we found that, indeed, most AI in surgical education studies were conducted in simulations. There was a focus on technical skills. And this means there's an opportunity for non-technical skill training, big time when it comes to artificial intelligence in surgical education. We also found the majority of studies used artificial intelligence to analyze data post-study. Only a minority used artificial intelligence to actually enhance training. So when we think about surgical education, most of that work with AI is really in the development phase. It's not at that kind of practical phase yet where students are coming in to participate in a study, students or residents, and they're leaving with value-added feedback from the AI. So this is, again, I think, an important direction. Also, educational theories are really absent in AI at the moment, although there definitely are theories, and I've been part of the artificial intelligence and education community for many years now, and it can be done. We can absolutely marry computer science, psychology, and education, and there's more work we can do absolutely in the surgical world and broader the medical world to kind of catch up. Some other gaps we have are that there's not been a lot of research in surgical education looking at expectations that people have for artificial intelligence, nor how training with regards to artificial intelligence can or should be done. These are important questions. The Royal College of Physicians and Surgeons released a report a couple of years ago indicating that people were very enthusiastic about working with AI and participating in it, potentially using it in clinics or participating in industry initiatives where AI is developed, etc. But we need to also have an appreciation of what people know, what people think they know, and where that motivation, confidence, comfort is coming from, and how we can build curriculums not just for emerging surgeons, but also potentially for surgeons who are practicing currently. As artificial intelligence itself is something that's generally being seen as an emerging competency within the medical fields. So that's something to keep an eye on too. We've done some work on knowledge mobilization with this topic. My former postdoc, Dr. Ilif Biljik, who's now an assistant professor at McMaster University, we published a piece in Conversation Canada, and also had a virtual panel on exploring the roles of artificial intelligence in health sciences education. You can listen to this talk where we had a number of participants from other universities, the Royal Colleges, former president participated, people from CAE, MedTech, etc. You can get to that via my website under completed projects. I think if you Google Sales Lab McGill, you'll find it. I wanted to provide an example of an AI-enhanced virtual reality sim to kind of bring some different pieces together. This is a project that was done by a student, Ali, who Rolando Del Maestro and I co-supervised together with Rolando's Neuro Sim platform, which is a really exciting platform. We were wondering whether feedback from an AI tutoring system compared with training by a remote expert instruction in learning, whether basically this would have an improvement in surgical performance. We did an RCT with 70 medical students, and we found significantly higher performance scores when training with an AI tutor compared with expert instruction and a control with no feedback. So that was quite interesting. We also found that students' cognitive and affective responses to learning with AI were similar to that fostered by human instructors. So this was a really, and Rolando's work generally has also been revealing some of the promise that AI has when combined with virtual reality. So that was fun work. Some of the things that's next, so as I mentioned, bringing AI and simulation together, there's a lot more we can do, especially when we think about moving beyond post-data collection performance assessment to try to give residents and medical students feedback on how they did, what they can improve on, and all the different metrics that we can glean using AI and digital tools really provide us with a rich opportunity to do that. So as I mentioned, we want to use that data to develop automatic feedback. This is how we can bring it in and give students feedback before they leave a lab. Is to have a strongly validated automatic performance scoring. So of course this requires strong validity evidence. We also want to move towards assessing multiple competencies. So as again the review noted, there's been a real focus on technical skills, which is normal, but we also want to look at the non-technical skills, and I would argue especially those that pertain to psychological well-being, which underscore other competencies such as communication, leadership, collaboration. If you're unable to control your emotions or stress, you're going to have a very hard time being a good leader, being a good communicator, being a good collaborator. We want this feedback also to be personalized. As I mentioned before, the artificial intelligence and education community has been doing work for a couple of decades, and some of my work is amongst this, looking at how we can personalize feedback, which is sometimes a little bit different than what feedback might look like from a human tutor, instructor, attending physician. And so getting a sense and running some studies on how we can personalize that feedback to residents, to medical students, and to people with different levels of prior knowledge in different places on that learning curve is important. We want, again, this feedback to be something that's ongoing and also responsive. And effectively, what I'm talking about is using artificial intelligence like a coach. So in other words, more than a non-playable character, but really moving towards something where the feedback is, again, ongoing, it's personalized, it's constructive, and it's evidence-based, it's effective. To get there, as I mentioned, we'll need high-quality assessments that are drawing on high-quality and diverse data, including both our channels and our sample. We want artificial intelligence to work for everyone, and that means we need diversity in the data and we need high-quality studies. The point is not to replace simulation individuals, so standardized patients, etc., but really to supplement what we can do in simulations. And some of the reasons for this is that with virtual simulations, we can offer potentially better availability and accessibility. Using screens, these are things that people might do at home. They might indeed use a virtual simulation before even coming in for that in-person component so that they can make the most out of that precious in-person simulation time. With that, and with things being online, there can be less fear of judgment or stigma, especially from failures that can occur. As safe a space as simulations are and are intended to be, doing something first online can further reduce that anxiety. We can also have an easier time accommodating and bringing in different modes. There's more flexibility, and also the diversity with virtual standardized patients, as that doesn't require necessarily hiring or purchasing a large roster of mannequins or hiring a large roster of standardized patients to represent different demographics of patients. And as we know, there's a very, diversity is multidimensional. And so if things are thoughtfully done and thoughtfully integrated, virtual standardized patients, could help improve diversity in simulation as well. So again, they're an opportunity to offer different and potentially valuable feedback as well to individuals and groups through various metrics. Again, there's all kinds of things that we can collect and score with virtual tools. And sometimes we also don't have as much time as we'd like with in-person simulation. So we can get different feedback, and in some cases, potentially even more feedback from what we do in the virtual. So I want to thank you so much for listening. If you have questions, please feel free to contact me. And also feel free to check out the great work that we're doing in my sales lab. Thanks so much.
All right, amazing. Jason, thanks so much for this amazing lecture. We now have about 10 minutes for questions. Do we have any questions from the audience?
Thanks, Julia.
Dr. Barlach?
Hey, Jason.
Hi, Jake.
Nice to talk there. Thank you. A couple of things. There are, for example, late types and early types. People that can't operate in the morning and people that fall asleep at 9 o'clock. Do you think there are stress types? And some people that can't get it together until there's a sufficient level of stress for them to focus, and other people who fall apart under stress?
Great question. An interesting comparison point with the time of days. What research has shown is that there's kind of a curve, and so people often perform optimally with some stress. So if we think about kind of a normal curve or normal distribution, a little bit of stress can be a good thing. It can be a motivating factor. It can remind and encourage people to to focus their attention on the task, take things seriously, etc. But if that stress curve moves too far up, this really it retracts from our cognitive resources. Instead of attending to performance, we start to instead worry about failure, which is really a change in terms of mind shifts. So where exactly that place falls can vary between people. And we would expect there to be individual differences with regards to people's ability to manage stress. And there can be, kind of individual background factors to that. But that's where learning how to manage stress can play a role too. So with some of the work,
I grew up in an old-fashioned British system where sarcasm and bordering on physical abuse in the classroom was unnatural physical abuse. Was pretty normal, right? And these days, people are very worried about inadvertently stressing people out too much. Do you think people can be trained to shift that curve, or do you think it's fixed?
Well, I'm sorry to hear about what sounds like a very unpleasant educational experience. And I guess the second point, I think that there is, and that's really the point of part of this program of research, is to equip people, with the tools to manage stress as it unfolds, particularly stressful or particularly sources of stress that we can expect to experience in clinical and surgical contexts. But you also gave me a really nice segue to talk about another related program of research that we're doing that's also funded by SSHRC, which is on anti-harassment. And that's to provide people with an understanding. We're focusing at the moment on residents and medical students of what harassment is and how they can understand this, and what they can also do in a constructive collegial manner.
Before you get into that, what I've always been wondered about this, what do you think is the motivation behind the harasser, right? Do you think the harasser is just to say this to like seeing people suffer, or do you think they have misguided honorable motives.
I don't think that it's Machiavellian sort of scenario going on. First, the harasser, as we've seen from research, can be anyone in a clinical situation. While some research is focused on the person with the most authority, the attending physician, for example, sometimes it's fellow residents, sometimes it's fellow medical students, sometimes it's the patient, sometimes it's the patient's family. It can unfortunately be anyone. And part of why harassment is such a challenge in healthcare context is because it's a high stakes context. We talked a few moments ago about stress and people's curves. Well, the type of stress people are likely to experience, in a typical office setting, for example, is really different from the type of stress we expect that people are going to be dealing with, in a high stakes context, such as an OR. And so when there's more stress, that is going to make it harder inherently for people to regulate their emotions. So it's really an environment where it's the perfect storm, unfortunately. And that's where it's so important to equip everyone involved with the tools to regulate their emotions, but also with the tools, speaking about harassment in particular, to recognize what's going on and to find constructive and collegial ways of managing that situation to ensure, again, patient outcomes. And so that's some of the work that we're doing.
Great, thank you.
My pleasure. Great questions, Jake.
All right, we're taking the next question from Dr. Fried.
Thanks, Jason. Great talk. And I really wanted to pursue that last area because I think that certainly in surgery, there are many situations that are highly stressful, like there would be in maybe emergency room, critical care, et cetera. But we're so cautious in our educational programs to avoid putting people in these situations that I wonder whether, in our concern about that, we are not preparing people as well as we should. And the transfer of training from a simulated environment to a real environment really needs to be able to mirror the type of stress and stakes that are there in the real world. I just wonder if you'd comment on how we kind of navigate that tightrope. We obviously want people to feel comfortable in the learning environment, but we also want the learning environment to prepare them for the discomfort they're going to have in the real world.
Really great question, Jerry. And it is true. One of the big advantages of simulation is not, and that's where I think we've tended to focus in the literature and also in our thinking about the technical skills. But managing stress is associated, as I mentioned, with many of our fundamental, CanMeds competencies, and that's where that needs to be important. And of course, technical skills can be impacted by our non-technical skills and by our ability to manage stress. So I would say that, again, we do need to do what we can to prepare learners and to provide them with the most authentic environments possible, so that they're going to, so they are prepared when they move out of those simulated settings. And that's, simulations are expensive. We don't necessarily have as much opportunity to do simulation training as we'd like. I think in some ways, this is where virtual simulation can play an interesting role to supplement what we can do with in-person simulation. I think that by using virtual sim to give people some of the fundamentals, to give them a little bit of practice doing things, that students and residents might be in a position where they can be exposed to slightly more complex, more urgent, perhaps more globally stressful situations in a sim than perhaps they are at the moment, because they would be a little bit further in their learning curve. Their prior knowledge would be improved, and their practice applying skills might be that much more developed. So it's definitely a bit of a careful needle to orient, but I think it's also necessary that we really find where that sweet spot is in medical student and resident training across programs, so that we give medical students and residents the best training they can have, and that this just advances the learning curve as much as possible, and also prepares them to be the most effective members of teams when they move into those clinical rotations. So complicated question, but I hope I've at least given you my direction of thinking on it.
Thank you.
All right, we have another minute. Maybe we'll take a quick question for Dr. Merwali.
Jason, great presentation as usual. So I was reading this book by Timothy Wilson, and it's entitled Strangers to Ourselves, Discovering the Adaptive Unconscious. So basically what it's saying is that there's a whole system that we cannot access. And this is basically due to evolution. So if you get a stressor, already the system is activated where you have fight or flight. So you have that system you can't access. And so how do you include that into AI?
How do we include it in AI in terms of the training? So one of the things
in terms of training, because that system is still running. It's evolutionally, it's protected us. You get a fast heart rate. It's to help you run from animals. But that system, you can't access it.
Great question, Faxon, thanks for that. So I would say that one of the things that AI can do, especially when it's combined and has access to physiological data, is it can provide, the wearer or the user with information, such as, I've noticed that your heart rate or your skin conductance has really started to climb or spike. Maybe we've seen an increase in the quantity of skin conductance responses, or we're seeing a sharp increase in the amplitude of that physiological response. And a suggestion, the AI through a virtual agent, might provide some suggestions in terms of whether that's just making the person aware that they're becoming stressed, so that they might take that into consideration when they're responding. It's easier to be more short, frustrated, and to, for example, to have responses that can be unconstructive to a situation when we're upset. So being aware sometimes of our own emotional state can be helpful. Also, if an artificial intelligence is able to identify that we're on a trajectory, to go back to what I was talking about in response to one of Jake's questions, if we're on this Yerkes and Dodds, trajectory where we're going far outside quickly of what's kind of an adaptive, helpful stress response, knowing that can also encourage us to kind of refocus on dealing or managing that emotion. And AI can even go as far as to provide suggestions about how, in a particular situation, that person might do something. Sometimes it's our ability, especially as we get anxious or upset, to think as level-headed or as clearly as we would as an external viewer can go away. So sometimes suggestions like that could also be helpful. I could talk a little bit more about that in more detail, but I hope that quick response provided a little bit of a response to that great question. And I think I forgot to thank Jerry for his question, which was very good before.
Yeah. Thanks, Jessica. Wonderful.
All right, Jason, once again, thank you so much for the amazing presentation. We're going to move on now to the student presentations. And just a quick reminder that the link to the presentation evaluation will be has just been posted in the chat, and the scoring for each presentation is identified by the name of the presenter. So our first presenter is McKenna Polk, and her presentation is entitled Patients' Preference, Attitudes and Beliefs Towards Receiving Cancer Surgery During the COVID-19 Pandemic, a Qualitative Study.
Hello, everyone. My name is McKenna. My pronouns are she, her, and I'm supervised by Dr. Giulio Fiore. Today, I will be talking about our work on patients' experiences undergoing cancer surgery during the COVID-19 pandemic. So in response to COVID-19, Quebec's Ministry of Health began repurposing surgical care infrastructure and delaying many non-essential surgical procedures. These measures quickly created a massive backlog of elective surgery cases, reaching over 70,000 within the first 2 months of the pandemic. Approximately 30% of these delayed cases were oncology cases, meaning that many cancer patients experienced delays or cancellations to their surgery. It's estimated that it will take approximately 2 years to clear this backlog of cases, with wait times returning to normal by the year 2024. However, delaying time-sensitive cancer surgery is known to decrease patients' long-term survival and increase patient anxiety and distress. In addition, cancer patients are already at an increased risk for COVID-19 infection and more likely to experience serious events. Thus, cancer patients are facing multiple health risks at the same time as managing changes to their care. Therefore, the objective of this qualitative study was to understand the experiences of patients undergoing cancer surgery during the COVID-19 pandemic. This was a qualitative description study conducted at the McGill University Health Center in accordance with CORRECT guidelines. Our study period was March 16th, 2020 to January 31st, 2021, which was selected to include patients who underwent surgery during the first and second wave of the pandemic. And patients scheduled for general cancer surgery were eligible if their surgery was performed on the date scheduled or delayed by the hospital's surgical repurposing measures due to a positive COVID-19 test or screen, or at the patient's request. Interviews continued until thematic saturation was reached, or the point at which no new additional interviews produced no new information. And a quota sampling method was used to ensure the recruitment of participants with a variety of different surgical and demographic characteristics. Our quotas can be seen on the table in the right. And finally, data were collected using semi-structured one-to-one interviews, which were audio recorded and transcribed verbatim. Data were analyzed using inductive thematic analysis informed by Vaughan and Clark, and this process proceeded in several stages, starting with interview transcripts, which were inductively coded, and then several rounds of iterative comparisons were used to identify sub-themes and finally themes. So between November 2020 and November 2021, a total of 20 participants were interviewed, and all of our recruitment quotas were met except for a slight underrepresentation of patients who were positive for COVID-19. Half of our participants were male, the mean age was 64 years, and the cancer sites represented were breast, skin, hepatopancreatobiliary, gastroesophageal, and colorectal. And 6 of our participants did not experience a surgical delay, whereas 14 did experience a delay either by the hospital at the patient's request or due to a positive COVID-19 test. Thematic analysis revealed 4 major themes, the first of which were considerations for prioritizing surgery. So patients assessed their willingness to still undergo surgery during the COVID-19 pandemic by weighing the risks of their susceptibility to infection, the healthcare system safety, and burden on the healthcare system against the risks of their health condition and the recommendations of their provider. For patients who did undergo cancer surgery during the COVID-19 pandemic, a number of changes and deviations from their expected treatment were noted. Those included the COVID-19 preventative measures, unconventional communication pathways, non-participatory decision-making, delayed and rescheduled care, alternative and interim treatments, and changes to the hospital environment. And these changes from the expected care did result in diverse psychological responses from participants. And these ranged all the way from satisfaction and a heightened sense of appreciation and gratitude for their care, all the way to a sense of injustice at having to undergo cancer surgery during the COVID-19 pandemic. In order to mitigate these responses and the distress experience, many patients employed adaptive coping mechanisms that included positively reframing interruptions to their care, focusing on mental and physical health, seeking additional information, communicating with their surgeon, and gradually acclimating to COVID-19 related changes. So in summary, delays and interruptions to surgical care did cause severe distress for many patients. However, consistent communication with the treating surgeon and transparency regarding COVID-19 related changes did facilitate acceptance of delays and protect against distress. These findings underscore the importance of proactively addressing patient concerns to foster trust and enhance communication. So in the future, as healthcare systems address the backlog of delayed cancer surgeries, providers should elicit patient concerns and initiate open discussions regarding the impacts of COVID-19 on surgical care to minimize patient distress. Thank you.
All right, thank you so much, McKenna. Just a reminder that for you guys to save your questions to the end of all the student presentations. So our next presenter is Amir Sayadi, and his presentation is titled Assessment of Accuracy and Repeatability of Augmented Reality-Assisted Epidural Needle Insertion. we can't see our screen. No, yeah, came up. Is everything okay there?
I think he got disconnected somehow. I have
okay.
his recording.
Maybe play
I'll play his
video.
recording.
Yeah, thanks so much, Nico.
No problem.
Hi everyone, this is my pleasure to present our paper entitled Assessment of Accuracy and Repeatability of Augmented Reality Assisted Epidural Needle Insertion. My name is Amir Sayedi, and co-authors of this study are Dr. Renzo Cecere and Dr. Amir Hushar. As a background, in this image and video, you can see epidural injection, which is a medical intervention to inject therapeutics directly at the vicinity of the spinal cord and the nerves branching from it. For epidural injection, a long thin needle is inserted into the patient's spinal cavities through the skin and underlying soft tissues. Technically, epidural needle insertion is a blind procedure that relies merely on the physician's tactile feedback. This tactile feedback can be polluted with needle tissue friction and may vary from patient to patient. Accurate insertion of the needle is a precise task that may result in irreversible damage to the nerves or spinal cord if not performed correctly. The robotic needle insertion can help physicians during the procedure to control the depths of insertion. As you can see, a serial robot with the needle insertion mechanism and needle driver attached at the end effector can help physicians by using a haptic input device to control the position of the needle inside the patient body. In our research, to enable physicians see the needle trajectory while inserting the needle, an augmented reality-based navigation assistant technology was proposed, and to generate patient-specific 3D holograms that were projected to the patient, to the physician's display. First, a patient-specific anatomical model was created based on a patient's lumbar CT scan using Mimix Medical materialized software, and the vertebral bone were extracted from the 3D model and was manufactured using 3D printing technique. To generate multi-layer soft tissue covering the vertebra, as you can see in the images, a multi-level molding technique was used using silicone elastomers. For augmented reality, Unity software and Vuforia AR engine used to localize the 3D model inside the real scene based on the position of the camera and a marker attached to the patient body. You can see in these images that this 3D model can help physicians to see the exact position of the needle during the procedure. For the proof of concept, the user repeated needle insertion with and without augmented reality assistant for 10 times. The accuracy of needle insertion was quantified by measuring the needle position error with and without assistance. Moreover, the repeatability was quantified by measuring the standard deviation of the position error. In this slide, you can see in this video the experiment and how the needle inserted inside the 3D printed model while the 3D virtual models can show the exact position of the needle inside the patient body and can help the physician to control the depths of insertion. Result shows that without assistance, the average needle position error accuracy was 4.3 millimeter compared to 1.03 millimeter with assistance, with 76% reduction. Repeatability 1.8 compared to 0.77. Success in entering the epidural space in the first try, 70% compared to 100%, and that's correct because the user can see exactly the positions of the needle inside the patient body, so they can control successfully entering the epidural space in the first try. And time to completion of one needle insertion, 16 seconds compared to 7 seconds. Of course, it depends on the speed of the driver, needle driver. That can be different, but with the same speed, the time for completion of one needle insertion was 16 compared to 7, with 56% reduction. To conclude, the proposed system improved the accuracy, repeatability, and success rate of epidural needle insertion on an anatomical model. It also reduced the procedural time. And the proposed system will be investigated in a broader user study that will include more comprehensive user experience and accuracy criteria. Thank you.
All right, thank you, Amir. I'm sorry about your technical issue.
Yeah, sorry.
So, yeah, the next presenter is Noam Swissam, and he'll be presenting on ultrasound nerve block guidance using deep learning.
Perfect. Just to confirm, can everyone see my screen and hear my voice?
Yes, it's perfect. Thank you.
Okay. So just 1 second. Okay, good morning, everyone. Thank you all for being here and taking the time to listen to my presentation. My name is Noam Swissam, and today I'll be presenting my research titled Ultrasound Nerve Block Guidance Using Deep Learning. So ultrasound-guided peripheral nerve blocks are used to administer anesthesia to certain nerve regions of the body to reduce the feeling of pain before or after surgical procedures. And these procedures make use of ultrasound feeds to guide a needle to a nerve of interest in order to inject anesthetic around it, effectively blocking sensation in that area of the body. And this is great because it offers continuous visualization of various anatomical structures, the needle and its path, local anesthetic delivery and its distribution, and peripheral nerve blocks in general avoid the need to prescribe addictive oral medication and induce general anesthesia. However, some limitations exist with these procedures that many studies point out. For example, they require extensive knowledge of sonoanatomy, leading to experience-related barriers. Limited experience can cause unwanted nerve, artery, or vein damage, local anesthetic toxicity, and in some rare cases, organ damage. And this can be further visualized in an image of the axillary brachial plexus. In this image, the radial, ulnar, median, and musculocutaneous nerves are all present and difficult to locate. And despite the lengthy and intensive training that anesthetists go through to perform these procedures, studies show that there are most often disagreements on sonoanatomy. And this brings me to my research. A deep learning software solution is proposed to address these limitations by providing anesthetists with a tool capable of locating nerve regions in real time via ultrasound. The study so far focuses on locating the transversus abdominis plane and the femoral nerve regions. The objective is to accurately segment the tap and femoral nerve regions in an ultrasound feed in real time. The hypothesis is that a trained deep learning algorithm can make predictions on the nerve's location with a minimum accuracy of 70%. For our methods, we are using a UNet architecture, which has gained a lot of traction in the biomedical field for deep learning tasks since it was first proposed in 2015. The reason being that it's very good at training a model on a limited number of images, which is usually the case in the biomedical field. UNets are capable of breaking down images into simple feature sets that the deep learning models could learn to recognize. In terms of data set sizes, the TAP training set contains 554 RGB images sourced from a YouTube video, and the test set contains 40 images. As for the femoral nerve data set, 562 images were found online and labeled. 500 were used for training, while 62 were used for testing. In our methods, we also use the common technique of augmenting our data set Augmenting a dataset is a technique used to artificially increase the size of your training set by introducing changes in the original images, effectively making them new, never before seen images. In this project, 5 augmenting techniques were used. And in addition to the 5, models were trained on no augmentations and all augmentations. And the following slides, I will demonstrate the augmentations. So just to note that the left column shows the original image and its mask, while the right one shows the augmented versions. Here in this augmentation, we have the flipping augmentation, which we flip the image horizontally. The rotation augmentation just rotates the image to a certain degree. The gamma intensity brightens or darkens the image, and this mimics the gain function on ultrasound devices. Then we have the elastic deformation, which adds a bit of warping to the image. And finally, we have the Gaussian shadow, which introduces a shadow randomly placed on the image with a Gaussian spread. And this can mimic air pockets that can sometimes be found in the body. So after having trained a model, all images in the test set were passed to make predictions on where the nerve is located. And using the dice score, a percentage on how well the prediction matches the labeled nerve was recorded. The dice score equation is calculated by taking the ratio between the true positive pixels and all other pixel categories. Lastly, the performance metric that was recorded was the mean dice score for the entire test set. So across all trainings for the TAP dataset, the model that was trained on the set containing the rotations received the highest mean dice score of 79.15%. And as for the femoral nerve results, the model that was trained on all augmentations received the highest mean dice score of 82.35%. And while the quantitative results are great for showing the training performance of a model, the qualitative results should also be considered as important. So in the next slide, a short video demonstrates the predictions of my TAP nerve in real time. So here we can see in green the predictions of where my TAP nerve is located, and to the naked eye, the predictions seem to be very successful. So our results have shown that it is possible to train future models to recognize other major nerve regions in the body, and have demonstrated advancements in accurately detecting the TAP and femoral nerve regions. Moreover, the UNet architecture has proven itself to be a great candidate for segmentation tasks in biomedical applications, specifically for ultrasound nerve block guidance. Future tasks might include adding new major nerve regions to train a model on, predicting the location of other anatomical structures in ultrasound feeds, and designing a novel graphical user interface to present all of our features in one place. A special thank you to my supervisor, Dr. Thomas Hemmerling, Dr. Chantal Stier, other collaborators, and the entire collaborative team at ITAG Laboratories. Thank you very much.
Thank you so much, Noam. And our last presenter is Tony Am, and he'll be presenting on mapping out conceptualizations of emotions in simulation-based education, a scoping review.
Hi, everyone. Can you hear me?
Yes.
Alrighty. Hi, my name is Tony, and I'm supervised by Dr. Jason Harley, and I'm here today to present a scoping review that our lab conducted entitled Mapping Out Conceptualizations of Emotions in Simulation-Based Education. The background for our study is that emotions are crucial in medical education due to their impact on learning processes and clinical decisions. This has led to increase in research efforts looking at emotions, but there's a concern regarding relatively weak theoretical integration. Weak theoretical integration can lead to misuse of terms, and studies can become susceptible to subpar methods of data collection and interpretation. To address this, we've conducted a scoping review with the following primary research question. How are emotions and closely related constructs, in other words, stress, emotional intelligence, and mood, conceptualized in articles that focus on simulation environments with medical students and trainees as the population of interest? We've decided to focus on conceptualizations of measurements of the constructs, along with looking at what kind of emotions were captured in the studies. For our methods, we've decided to receive guidance from Arksy and O'Malley's methodological framework, along with the Prisma SCR checklist. We've also received consultation from our health sciences librarian. Shout out to Andrea. Some key decisions for our review include us deciding to use 2 databases, Medline and Embase. Our search was carried out in 2020, and it covered the past 10 years. We've also decided to hand search 2 relevant journals, Medical Education and Academic Medicine. Further, we ensured minimum 75% inter-rater agreement rate for screenings and extractions, and we've went through multiple pilots to ensure quality. We've conducted descriptive data analysis to answer our research question. This diagram shows the overall picture of how our project went. We began with 29,000 articles, and after deduplication, a title abstract screening, and 2 rounds of full text screenings, we ended up with 139 articles. With the addition of 2 articles from the hand searching, the total articles we had for our review was 141. For the results, we can see here that mood was featured only once. Emotional intelligence was also featured less relative to emotions and stress, which each of them being featured 34, 45, and 88 times respectively. I'll quickly note that for this graph, articles can feature multiple constructs simultaneously. So articles featuring both stress and emotion will contribute to both of the counts. When it comes to examining whether studies formally defined these constructs or not, articles that featured emotional intelligence had 65% rate for having a formal definition towards what the articles were focused on. Stress articles had much lower rates with 27%, but articles dealing with emotions had the lowest rate with just 21%. For measurement types for emotions, the majority of the articles relied on just self-report. In fact, it's worth noting only one study used both self-report and physiological measures. For measurements of stress, while self-report instruments were common, just like emotion articles, other types of measures were also common, notably measures related to cortisol and heart rates. Finally, for when looking at the types of emotions medical students and trainees experience, we saw anxiety was the most frequently captured emotions, with 25 articles focusing on them. Fear was the next most common, with 10 articles focusing on them. To conclude, it seems on the surface that medical and surgical education is dominated by fear and anxiety. And while this may be true, it's important to be mindful of what these studies' goals were. Many studies set out to measure nothing but anxiety, and with instruments like the State Trait Anxiety Inventory, the goal of the study isn't to capture a wide array of emotions. The studies that wanted to capture such negative emotions also make sense, since simulation-based education often deals with scenarios that are meant to elicit negative emotions, such as simulation of a patient death. We also found that the studies lacked integration of theory, particularly when dealing with emotions. We believe this contributes to lack of guidance on measurements of emotions via multiple channels, or perhaps on what emotions researchers could look for in the first place. We believe that studying other types of emotions, such as guilt or pride, can support enhancing surgical practice and training. One limitation of our study was that we did not evaluate the impact of these emotions in simulation-based education environments. Naturally, a future direction is to conduct a systematic review that looks at just that. Here are the references, and thank you for your time.
All right, thank you so much, Tony. So guys, we have time for one question or two. Do we have any questions from the audience? All right, Jason, do you want to start?
Thanks so much. Great presentations, everyone. Makina, I had a question for you. Really interesting qualitative study. And it nearly seemed like you might explore, depending on where this is with the publication trajectory, it sounded nearly like it might be a grounded truth study, and that you're trying to maybe build a theory of how these different components connect. But I wondered, you talked about coping strategies and presented some of the strategies that were there for adaptive. I wondered if you found any evidence of maladaptive coping strategies, as these are also common, and I wondered if they came up in any of your interviews with patients.
Mm-hmm. They didn't come up super frequently, not enough to derive like an entire theme out of it, but there were definitely several participants who were ruminating over the delays and the changes to their care. A lot of what we found was related to sort of death anxiety and as well as concerns regarding progressive disease. And so there were certainly some participants who were sort of ruminating over their distress and their anxiety related to that. What we did find was that participants who felt that they were adequately informed and found that information dissemination was quite transparent we're better able to employ those adaptive coping strategies, and we're protected from a lot of sort of the distress and concerns that other patients experienced. I'm not sure if that answered your question.
It does. Thank you. I'll pass the question on to someone else so we don't have a lot of time. Great work.
Julia, I'm not sure if you noticed the question in the chat for McKenna.
McKenna, yes. So one question from Katie McKenna. Did you distinguish between surgeries delayed to the patient's positive COVID test or the surgeon's positive test? If so, were there differences in responses? Sorry, I think you're muted, McKenna.
Sorry about that. Katie, just to clarify, do you mean the surgeon testing positive for COVID-19 versus the patient testing positive for COVID-19? Or do you mean the surgery delayed because of the repurposing measures?
Sorry, let me clarify. So was the positive test cause of delay for a patient's positive test or for a surgeon's positive test, or could it be either?
Oh, okay, I see. It was for the patient, specifically either receiving a positive COVID-19 test or experiencing symptoms of COVID-19 and therefore having their surgery delayed. So we had a couple of those participants, and there were some distinct characteristics of those participants who did have a positive COVID-19 test. Something that was really interesting and unexpected finding that came up is actually they had a little bit less anxiety and concern related to the hospital environment and their susceptibility for COVID-19 because they believed they had some immunity to the virus and were less concerned about the potential of infection because they'd already experienced it. So that was an interesting finding that I didn't really expect to see, but would be interesting to look into further because those participants were a little bit underrepresented in our sample. Thank you for your question.
Jake, do you have a quick question?
Yeah, quick question for Amir. You presented us some, fairly impressive time error reductions or improvements in accuracy. I would like to know which, or if any of them are clinically relevant. Like you went from 16 seconds to 7 seconds needle insertion. Does that really shorten the needle insertion? Because I think the preparation is maybe 4 or 5 minutes, right? And so which one of those is the most important clinically?
Thank you for the question. First, apologize for the technical problem happened. And I want to mention that our study was a proof of concept of using the needle insertion because the testbed was ready, but we are aiming to use it for the cardiac models that we need sequential CT scans for generating the 3D models. But for this study, for preparation, of course, we compared the time when users start inserting the needle inside the 3D model or 3D printed model. So we compared the time when they start. So both are using the robotic system. So the needle is driven by a motor. So the time that they are inserting this needle inside the model were compared when they, because they don't, when there's no assistant, the user cannot see the position of the needle inside the model. So they just think that the needle is somewhere inside the model, so they can feel by the haptic device or just by the experience. But when the user is using the AR assistant, because they can see exactly the position of the needle inside the model, so they can go faster, so they can drive faster, and they can stop precisely exactly at the time at the position they want. So the other time that is for the preparation of the this insertion is not compared here, just the time that they started to insert the needle and the stop time that they thought that it's the correct position. And then we measured the penetration depths and found the error, the accuracy of the needle inserted inside the 3D model.
Great, thanks.
All right, thank you, Amir. Guys, I'm going to wrap up here because we're a bit over time. Thanks everyone for the great presentations. Faxon, I'm not sure if we want to stick to like the 5-minute breaks or just continue directly to the breakout rooms.
Yeah, I think we should just continue to the breakout rooms. Is that at 11:50? Unless anybody would like to take a 5-minute break. What do people think?
Right.
Right, okay. 5 minutes break then, 11:55, is that?
Cool, that's perfect.
Okay,
all right.
11:55.
Thanks so much, everyone. Cheers.
Yeah.
Maybe Noam, you could answer the question in the chat.
Yes, absolutely.
Thanks.
Sorry, just sorry, Shrita, do you mind clarifying your question?
Hello?
Yes.
Yeah, sure. I was just wondering what's the current like gold standard for guiding for tap blocks, and whether you were able to compare like your ultrasound guided deep learning technique to it, and like if it was considered more beneficial than the current technique.
Right, absolutely. So as of now, well, before they used nerve stimulation to guide needles to perform these nerve blocks, and as of late, ultrasound guided peripheral nerve blocks have been the new gold standard to perform these techniques. As for using deep learning and machine learning, it's pretty novel. There are a couple of papers out there that I've been comparing to. That is why I chose the 70% benchmark to compare my results with, and the 70% is what all those studies have and all those projects have achieved so far for different nerve regions, specifically the femoral nerve axillary, brachial plexus, and the tap. So did that answer your question?
Yeah, thank you.
Time to start. Okay, excellent. All right, I'll be the chair of this session now for IRR Experimental Surgery Day. I'm Derek Rosenzweig, Assistant Professor in Department of Surgery and Junior Scientist in the Injury Repair and Recovery Program at the Research Institute of McGill University Health Center. So it's my pleasure to introduce our next keynote speaker, Professor Rita Kandel. Dr. Kandel obtained her medical degree and did her pathology residency training at University of Toronto. She did a postdoctoral fellowship at Tufts University and Massachusetts General Hospital, the other MGH. We're at the Montreal General Hospital. And she's a clinician scientist and currently the chair of the Department of Laboratory Medicine and Pathology in the Timurtri Faculty of Medicine at the University of Toronto, and the medical director of Departments of Pathology and Laboratory Medicine at Sinai Health System and Women's College Hospital. Her research is focused on utilizing regenerative medicine approaches to develop treatments for joint disease and intervertebral disc degeneration. Her research program has been supported by grants from CIHR, NSERC, NIH, and the Arthritis Society. She has published over 240 papers and multiple book chapters. Among other honors and awards, Dr. Kandel is a fellow of the Canadian Academy of Health Sciences. And today she'll be discussing biological cartilage repair, getting there. Thank you.
Well, thank you very much for that kind introduction. Can I share the screen?
Somebody should have given you permission. Okay, there you go.
Can you see it? Perfect, perfect. So it's wonderful to be here, even though it's around a Zoom table, and the presentations I've heard to date have been really superb, and I'm looking forward to this afternoon's presentation. So thank you for including me in this day. I thought today I might give you an overview of our quest to develop a method to repair cartilage. And we've known since 1744, thanks to William Hunter, that adult articular cartilage, once it's damaged, will not repair itself. And it really wasn't until the seminal work by William Green in the 1970s, in which he was able to show that you could isolate chondrocytes from articular cartilage and grow it in culture, that we dare to start talking about biological repair of the articular cartilage. So since that time, we have had numerous investigations in this area and a lot of work done. And if you do a search in PubMed under the term cartilage repair, you'll see approximately 17,000 publications. So clearly we've got a much deeper understanding of this process. But unfortunately, if you then do a search in the clinicaltrials.gov website, you'll see that there are 43 ongoing clinical trials, which will then tell you that we haven't yet identified the optimal way to repair cartilage. And you might ask, why is that? When you look at cartilage, and this is what it looks like down the light microscope, you will see tissue is composed of chondrocytes surrounded by large amounts of this blue staining extracellular matrix and a layer of calcified cartilage, which integrates with the underlying bone. And it does look very simple and straightforward morphologically by light microscopy. There are no blood vessels, there's no nerves, there's no admixture of cells, there's just the chondrocytes. And we know the composition of this tissue is predominantly proteoglycans, aggrecan in particular, type 2 collagen, water, but there are also other molecules present. And interestingly, these molecules, all of them, are differentially expressed in articular cartilage, such that it induces a depth-dependent zonal architecture. And you can see that in this figure that I've borrowed. And you can see that in the superficial aspect, there's less proteoglycans and more collagen than in the deep aspect of cartilage. And the other molecules that are present in smaller amounts, like PRG4 or lubricin, is predominantly located in the superficial aspect of the tissue. And alkaline phosphatase, an enzyme involved in mineralization, not unexpectedly, is localized to the deep zone of cartilage. And it is this differential distribution of matrix molecules and this zonal architecture that has been very difficult to replicate, both in vitro and in vivo. And that has been the major limiting factor that is impeding clinical translation of cartilage repair methodologies. So articular cartilage is very complex, and it's not surprising that it's not repaired, cannot repair itself. But there is a great interest in biological repair. And currently there are 3 methods that are being used around the world. And the one that really caught our eye was the autologous chondrocyte implantation method. And the reason it did was because it uses chondrocytes. This was a method that was described a number of years back and developed by Dan Grandy, Lars Peterson, Mats Brittberg, in which they took a biopsy of normal cartilage, isolated the chondrocytes, expanded them in cell culture, passaged them twice, harvested the cells, and then injected them into the defect under a patch. this methodology, which uses chondrocytes, one would think would be the optimal way to get repair of tissue that resembles normal articular cartilage. However, when they biopsy this repair tissue, as you can see here in this histological image, the morphology of the repair tissue is quite different than that of normal healthy cartilage. And actually, these cells synthesized a fibrocartilaginous type matrix in most cases. And fibrocartilage has limited weight-bearing capabilities and will start to degrade in about 7 to 10 years. So clearly, ACI, although in principle should work wonderfully, didn't seem to. So we wondered if we could understand what was going on with these cells in this process of culturing, that we could perhaps identify the conditions that would be needed to induce these cells to produce the right kind of matrix that you could repair articular cartilage. So Justin Pereno, a PhD student in the lab, took on this project. And Justin now has his own research lab in the States. Very proud of him. And what this project involved was taking bovine cartilage, harvesting the cartilage, isolating the cells, and plating them in monolayer culture. And you can see here that these isolated cells are round. And if you examine the cytoskeleton, you'll see that the filamentous actin is present cortically in the cell, and there seems to be this segmented distribution of globular actin. Now, if you passage those cells in monolayer twice, similar to what we were seeing in, it was what occurs in ACI, you can see that at passage 2, the cells have a very different morphology. They're more spread. And if you examine the actin cytoskeleton, you will see that the cells have lost the cortical distribution of filamentous actin. Instead, you can see actin fibers throughout the cell, and you can see the presence of stress fibers. And there is very diffuse distribution of the globular actin. So clearly there are very profound changes in the cell cytoskeleton. Concomitantly with this, the cells decrease their expression of the proteoglycan aggrecan and collagen type 2, the two major macromolecules in cartilage, and simultaneously start to increase the expression of collagen type 1. And so it's not surprising that these cells produce a fibrocartilaginous type of matrix because of this genotypic change. He also noted that if you plated these passage cells in a 3D membrane culture, and this was a method that we've developed that promotes cartilage tissue formation by primary cell chondrocytes. As you can see here, these passage cells lost the ability to accumulate large amounts of proteoglycans. So clearly, these cells had lost their hyaline chondrogenic phenotype and become more non-chondrogenic or dedifferentiated cells. And this explained why, although we were using chondrocytes to try and repair the defect, we weren't getting hyaline articular cartilage reproduced in this condition. So we asked the question, could we identify the culture conditions that would allow these cells to redifferentiate, reacquire their hyaline cartilage or chondrocyte type phenotype, and regenerate actually the appropriate articular cartilage? Went through the literature, and we identified TGF-beta as a growth factor that played a role not only in pushing cells to chondrocytes during development, but also in maintaining articular cartilage postnatally. So this seemed to us as a very important molecule and one worth exploring. And Vanessa Bianchi, a PhD student in the lab at the time, and who's now a research associate, took on this very question. And in this series of experiments, she actually used human cells, human chondrocytes obtained from both low-grade osteoarthritic cartilage and high-grade osteoarthritic cartilage. And we wanted to use human cells because obviously clinically, that's what we would use ultimately. So we wanted to be sure that we could actually push these cells back to what we would consider articular cartilage type chondrocytes. So she harvested the chondrocytes and processed them in a way that is standard. She put them in monolayer culture, passaged them twice, and then put them in our 3D membrane culture to try and grow them in the presence or absence of TGF-beta. And we had done a series of experiments to determine that this concentration was optimal. And what she was able to demonstrate is that in the presence of TGF-beta, these cells could be induced to produce a very robust cartilage tissue, as you can see here macroscopically. And when we evaluated its composition, it had the characteristics of hyaline cartilage. There was abundant type 2 collagen, abundant aggrecan, and there was no collagen type 1 present in the tissue. Interestingly, we could also demonstrate the presence of collagen, which in the superficial aspect was oriented parallel to the surface, which is a characteristic of superficial zone chondrocytes. And along with this, we were able to demonstrate a differential distribution of some of the macromolecules in cartilage. For example, we demonstrated that PRG4 or lubricin was present in the superficial zone of this tissue. Similarly, clusterin, which is another superficial zone marker, was also present in the superficial zone. So not only were we getting robust cartilage tissue, it was also showing some degree of zonal architecture. So we wanted to understand a little bit about this process of TGF-beta and its effect on these cells, because we wondered if this might actually be useful not only in understanding the process of redifferentiation, but as identifying potential biomarkers. And so we know the literature is really replete with investigations relating to TGF-beta signaling. We know that it binds to receptors and co-receptors are involved. But the literature suggested that if it bound to the ALK5 or the TGF-beta receptor 1, that these signaling that would be activated would push the cells towards a chondrogenic pathway. So we chose to look at the levels of ALK5 and the co-receptors TGF-beta 3 and endoglin in our study to see what kinds of changes were going on in these cells when they were exposed to TGF-beta 3. And we got some very interesting results. I'm not going to show the receptor 3 results. I'll just, for time's sake, I'll show you the ALK5 results. And we are comparing its expression in chondrocytes that were never passaged and can't make tissue. And this is characteristic of human chondrocytes. Even primary human chondrocytes do not make cartilage tissue in culture. But the passaged human chondrocytes grown in the presence of TGF-beta where it could make tissue. And what we could show by flow cytometry is that in both conditions, the chondrocytes expressed ALK5. And interestingly, the amount of ALK5 present on the cells did not vary whether it was P0 cells or P2 cells, or even whether it was cells from low or high-grade osteoarthritis. We saw similar effects for the R3 protein, which was expressed in very, very low amounts. Less than 5% of cells were positive. But what was really intriguing, and I think Annie Phillips, if she's here, will agree, were changes in endoglin. And this is one of the co-receptors. And what you can see is that the passage cells, which can make tissue now, express high levels of endoglin. So most of the cells express endoglin, and that the amount of endoglin per cell is much higher than in those cells that can't make tissue. And this is just the image of the cells. And of course, the density was corrected for the size of the cells, which are larger in the passage cells. So clearly, endoglin appears to be important in the process of regulating cartilage formation by passage chondrocytes that had redifferentiated. And just to confirm this, Vanessa went on to knock down endoglin to determine its effects on tissue formation by these passaged chondrocytes. And so what she did was she transfected the cells with siRNA against endoglin, and as a control, a scrambled RNA. And as you can appreciate, her knockdown was able to decrease the expression of endoglin for a prolonged period of time. And when we looked at protein levels, there was also a decrease in the amount of protein. But most importantly, when she plated these cells with a knockdown in endoglin, she could demonstrate that these cells had lost their ability to form cartilage-like tissue at 7 days, and this was actually maintained to 14 days. And just to allay any fears, this was not a result of differences in cellularity, because when we quantified DNA content, it was similar in the tissues that were formed by cells, which endoglin had been knocked down, as compared to the cells in which endoglin had not been knocked down. So clearly, endoglin is critical to the formation of tissue by these redifferentiated cells. It may be that ALK5 is also part of this process, because these cells all were expressing ALK5 as well, but endoglin is definitely critical to this process. It also seemed to us that we now identified a marker that we could potentially use to identify cells that would have the ability or not to produce tissue when they were redifferentiated. So we were obviously very excited about this, and Vanessa went right ahead and did an animal study to evaluate the ability of these cells to repair a cartilage defect. So she used a rabbit model, isolated rabbit articular chondrocytes. She passaged them in culture. And some of the cells she put into 3D membrane culture, which induced redifferentiation of the cells. And some cells she used directly, and these were the control cells. These cells that were in membrane culture and formed cartilage tissue in the presence of TGF-beta. She isolated the cells and then either mixed the redifferentiated cells or the dedifferentiated cells in this fibrin clot, which you can see here. We'd also already previously demonstrated that this fibrin clot was able to maintain phenotype, so we weren't worried about adverse effects of the carrier. She went ahead and implanted the cells into the defect, which you can see right here in the rabbit knee joint. And when she examined the repair tissue 6 weeks later, she got some very interesting results. She was able to demonstrate that in the rabbit defects that were repaired with the dedifferentiated passage cells, she could induce fibrocartilaginous repair, which was similar to what we see in ACI. And is just to point out, that's very different than the histology of the normal joint. What was intriguing was that the redifferentiated chondrocytes produced a granulation tissue type repair, which was entirely unexpected. And you can see that macroscopically that the cells are, the repair tissue is very vascular because it's very red. Now, this is a 6-week study. It may be that these cells somehow undergo remodeling and form chondrocytes. But at any rate, we decided that perhaps redifferentiating the cells was not going to be the answer. So instead, we said, well, we've got a method in which we can generate tissue. Why are we implanting cells? Why are we not implanting tissue? So we went back to the drawing board and tried to answer that question. We isolated our chondrocytes, formed our tissue, and this again is an experiment that was done using rabbit articular chondrocytes, and harvested the tissue and implanted it in a full thickness cartilage defect in the rabbit knee, and waited patiently until we could examine it histologically. And this is the histological appearance of that defect, which you can see here. And you can appreciate that the tissue is not affixed to the bone and is sort of floating free. And we realized that the next problem that we had to address was fixation of this tissue into the defect. Because we know from other studies that movement or micromovement impairs integration. So we went back to the drawing board and said, can we create a better way to fix these tissues into the defect? And we took a lesson from the osteochondral transfer approach, in which osteochondral plugs are placed into a defect, and asked whether we couldn't make our own osteochondral like constructs in vitro that would get away from all the problems of using autograft tissue. And so we went ahead and tried to do that. So together with my collaborators, Bob Pillier and Mark Grimpus, we worked hard on identifying a bone substitute, because that was going to be the first issue we needed to address. And so using calcium and phosphate, we were able to sinter these materials and develop this porous, biodegradable bone substitute, which has strength similar to bone. And when we implanted it into bone, as you can see here, the biomaterial is white, and the bone, which is stained black, is growing through the porous component of the material. You will note that there is no reaction to this material, which suggests that this is a really optimal biomaterial to use for this. We then went and asked the next question, which is, can this biomaterial support tissue formation? And so we did our usual, created our bone substitute, seeded it with cells, grew it in culture, and generated what we are calling this biphasic construct, which is composed of a layer of cartilage tissue integrated to the underlying bone substitute, and the tissue cells grew into the upper third approximately of the bone substitute, so it's well integrated. Characterized this tissue, and it had hyaline cartilage features. And so we were pleased that we had developed an osteochondral-like construct. So we went ahead and implanted this into sheep knees. We created a defect, put in our construct, and then waited up to 9 months. And this is the appearance of these constructs at a 3-month harvest point. You can see the 2 implants in the sheep knee, and you can appreciate the bone substitute material here on the X-ray. And when we examined it histologically, this is what we saw. So this is native cartilage, native bone. This is our implant. This is native cartilage, native bone, as you can see schematically here. And I think you can appreciate that the articular cartilage is in the right place. It's intact. It's integrated to the adjacent native cartilage. And it's hard to appreciate the bone ingrowth that's happened in here, but the implant is fixed in the into the defect. And if we just implanted the CPP alone, we got a fibrous layer overlying the CPP. So clearly very different than our implant. At 9 months, the tissue is still intact. And again, there was integration with the cartilage and the bone. And very interestingly, when we harvested this and characterized the tissue, we found that the weight-bearing capability of this tissue increased 20-fold over the 9 months of follow-up. So clearly this tissue has the ability to remodel and improve its weight-bearing characteristics. Well, of course, as you can imagine, we were really very excited and said, okay, this was in the trochlear groove. We now need to put it in a weight-bearing location in the joint, and we need to scale up the size because we were using a smaller size implant. Sheep knees are bigger. And so we went ahead and did that experiment. We again used sheep knees. You can see the defect here. There's the implant going in, and this is what the implant looks like after we placed it into the defect, sheep knee. We closed up the knees, let the sheep ambulate and walk. And 3 months later, harvested the implants. And here we are. Here is the macroscopic appearance of that implant. And we were actually quite disappointed because many of the implants, the tissue had sheared off. In other areas, it was, in other implants, it was fragmented. And we weren't expecting this result, but when we took a step back, we thought, well, there's probably 2 issues here. Could it be that the tissue just wasn't strong enough in that first stages of weight bearing because the sheep could freely ambulate after the implant? Or maybe the tissue interface with the bone substitute was insufficient. So we tackled both problems. So the first thing we did was, could we enhance cartilage strength? And knowing what we knew about the improvements with weight bearing, we thought the easiest way to do this would be to mechanically load the cartilage in vitro. And I won't burden you with all the details, but we looked at time, duration, length of culture. And what we ultimately landed on was applying cyclic compression for just 30 minutes, 24 hours after seeding. And that stimulation, not 15 minutes, not 60, but 30 minutes, was able to increase the synthesis of the matrix molecules, collagen and proteoglycans by 50%. And when we examined the tissue 4 weeks later, the amount of proteoglycans and collagens were significantly greater than the unstimulated or unloaded cells. And importantly, the weight-bearing capability, the equilibrium elastic modulus, had doubled. So clearly, we have a way to condition the tissue and make it stronger if we need to. So we had solved that problem. But we knew that the real problem was this interface between the cartilage and the bone substitute, because we know that there is a zone of calcified cartilage in articular cartilage, and that this zone of calcified cartilage is critical in the transmission of load across the joint. And it has been speculated that in this zone of calcified cartilage distributes forces laterally and absorbs some of the forces that come across the joint and prevents the shearing of tissue off bone. Because cartilage is soft, bone is hard, and this is the interface that protects that integration. So can we generate a cartilage with a calcified layer? Because that was probably going to be critical. And again, just to summarize the PhD project of David Lee, who is actually went into industry. It was his PhD project. We found that we couldn't use chondrocytes because when you passage chondrocytes, they lost their ability to undergo mineralization. So he developed a methodology that utilized mesenchymal stromal cells. And it was a two-step process that involved the differentiation of these cells in a membrane, standard differentiation in TGF-beta. And then he harvested some cells from this cartilaginous tissue, plated it on the bone substitute, and cultured them for 3 days in the presence of beta-glycerol phosphate, a known agent to induce mineralization, as well as a thyroid hormone T3. And what he was able to show is that that T3, thyroid hormone, induced alkaline phosphatase activity in these cells, which we know is important for mineralization. Then he went back, harvested more chondrocytes from a different set of redifferentiated or differentiated cells, seeded it as a second layer on the bone substitute cartilage tissue. And this is what he was able to generate. I'm going to say that this was a number of years of work. And what you can see here is our construct with the cartilage tissue integrated to our bone substitute. And when we did a micro CT, I'm hoping you can see a thin layer of mineral that is adjacent to the bone substitute, which you can see here. And when he examined it histologically, you can see a layer of hyaline cartilage, a layer of calcified cartilage integrated to the underlying bone substitute. And when he took this tissue off the cartilage, off the bone substitute, the cartilage tissue, which you can see here, is rich in proteoglycans, has a layer of mineral, as demonstrated by the Boncossa staining, is rich in collagen type 2, which is what we would expect, has a layer of a deep zone layer, which contains collagen type 10, another collagen present in mineralizing tissue, and has no Col1. So this is hyaline cartilage with a calcified layer. And most importantly, when he characterized the mechanical properties, what he could see is that the weight-bearing capabilities increased and that the strength or force required to shear the tissue off had increased 5-fold. So clearly he had constructed a tissue that was now osteochondral-like with hyaline cartilage, mineralized layer, and then we had to test it out. I'd like to say there's a drum roll. There isn't. This preclinical evaluation is now ongoing, and we're all going to have to wait another couple of months to get our answer. But we're very hopeful that we have solved all of the issues that we think are critical to proper cartilage repair. So in conclusion, I hope I've shown you a biomimetric approach to cartilage repair that involves cells to cartilage tissue or osteochondral-like constructs, and the importance of preclinical trials to evaluate all of the things that you're doing. And hopefully, next time I meet you, I'll have great results to tell you. Just want to acknowledge all of the people that were involved in this study and the funding organizations that supported it. Thank you very much.
Thank you, Dr. Kandel. This was a very nice story you told, clearly encompassing many years of work. I'll open this up now to the floor for questions for Dr. Kandel. If you have a question, just take yourself off mute, please, and ask your question. Okay, while they're thinking, I'll start off with a question. So it's quite interesting that, you know, a number of years ago, Athanasio's group did a nice study comparing TGF-beta 1, 2, and 3 on chondrogenic impact. I don't remember if it was on stem cells or on primary cells, but clearly there's multiple isoforms, and you chose to go with TGF-beta 3 as your differentiation factor. Do you want to comment on that and perhaps give us a little bit more rationale why you went down this pathway?
Absolutely. So thank you. We actually tested out TGF-beta 1 and TGF-beta 3, and we found actually that TGF-beta 1 was not reliable. And so it didn't consistently induce this redifferentiation process, whereas TGF-beta 3 did. And that was the reason that we went with 3. We didn't actually evaluate 2, and in part because the literature suggested that differentiated chondrocytes probably may not recognize TGF-beta 2. It is more a factor that's involved during development. And that was the rationale.
Thank you. Any questions from the panel?
Sure, I have a question. Thank you, Dr. Kandel. Very impressive, actually. I really enjoy these plugs, cartilage plugs, and the formation of bone is actually what I'm kind of curious about. I thought that was very, very impressive. And I was just, the question would be, so you see the formation, you see I guess the transition of chondrocytes going from, I guess, presenting collagen, expression of collagen type 2, and then as you go down the zone, you start forming the, I would imagine you have hypertrophic chondrocytes that would appear with the expression of collagen type 10. And during that process, have you also seen any differentiation to like osteoblasts? Like there is, I guess, a thought that some chondrocytes can differentiate into osteoblasts. And I was wondering if that is also taking place, and as why you see the formation of calcified cartilage in those lower layers.
Yeah, so we were actually interested in that as well. And so we could never identify expression of late differentiation. So for example, the cells were negative for expression of osteocalcin. So it really did seem like it was stopping at the early stages of mineralization. What will happen when we implant it? We're about to find out. But in vitro, we didn't see any evidence of differentiation towards osteoblasts.
So I guess it's possible when you implant it, which would be more interesting,
Mhm.
if it recruits, like you mentioned, yeah, I guess you're alluding to the recruitment of osteoblasts, as I think hypertrophic chondrocytes were also predicted to do some sort of recruitment from the, yeah.
Yeah, yeah. But it's interesting, you know, chondrocytes in cartilage, it mineralizes, but it doesn't ossify. So it has to undergo resorption and remodeling before that happens. So yeah, there's something interesting about cartilage that I think articular cartilage in particular that prevents that process from happening. And I don't think we know enough about what regulates the mineralization of cartilage, because it doesn't lose the aggregate expression. It doesn't lose the Col2 expression, as can occur in growth plate. So it's an interesting, it's similar but not identical to what happens in the growth plate.
Yeah, in growth plate, they undergo apoptosis as well, I believe, right? If I'm not mistaken. Yeah, and here they don't, I guess you're not seeing any apoptosis or apoptotic markers in those chondrocytes.
Well, we do see some, but not a diffuse, we don't see all the cells undergoing apoptosis. Actually, you can see chondrocytes within the mineralized tissue. You're right.
Thank you, Michael. Dr. Philip is next.
Yeah, thank you very much for a very amazing talk. It was very interesting. Enjoyed it very much, especially the Yandagun story. We had tried to tackle it, and we used the heterozygote mice. Of course, the homozygous are lethal. And after when we did the DMM surgery, we didn't see a difference in terms of repair. But in vitro, we have fantastic results. I'm also, in terms of ALK1 versus ALK5 pathway, ALK1 is the bad pathway, ALK5 is the good pathway, or chondrogenic pathway. So we had tremendous differences in vitro, but we were really disappointed when we didn't see that, at least in the heterozygote mice. We never explored the homozygous knockout embryonic, I think we did not do that. With siRNA in vitro also, where siRNA knocking down endoglin, we had again good results, but again goes to show that the in vitro results could be fantastic, but in vivo, when you test it, it didn't pan out. Now, I was also intrigued by the beta 1 versus beta 3 story. You said you had found the inconsistent results with the beta 1. Is it in terms of collagen 2 synthesis, or did you try looking at say MMP13, ALK1 versus ALK5, those kind of at the molecular level?
So I'm sorry, I didn't hear the question in there.
The question is whether, in terms of making a distinction between beta 1 versus beta 3, did you look at the molecular level? And were you looking at just collagen 2 expression? What were the endpoints that you were studying?
So the endpoint of the TGF beta?
Beta 1 versus beta 3, to make that distinction, you said that beta 3 appeared to be more consistent in terms of which endpoints?
Making cartilage tissue.
Cartilage tissue.
Yeah.
Okay.
Yeah.
So I was wondering whether you looked at parameters like MMP13 or ALK5, ALK1.
No. We didn't. And if I were to do those experiments again, I would want to look at the mechanism for the difference, because I'm sure it would provide us insight into understanding the process. But we didn't. Our endpoint was really, could we get these cells to redifferentiate and make the tissue that we wanted them to make? And we stopped there.
Yeah. So they are known to have differences. Beta 3, at least in the skin literature, is antifibrotic, opposite to a beta 1. So I was just wondering, we never really tested the differences between those two on chondrocytes. We should perhaps do that to see.
Yeah.
Okay, very interesting.
So I'm going to say that your work on endoglin inspired us to look at that molecule, by the way. just so you
No, we
know.
were intrigued that when we first found endocrine on chondrocytes, we were excited and told Van Dyke when I met him at a meeting, a better meeting. I said, "Oh, we found it in chondrocytes." He said, "No, it is all, and it's only on endothelium." So, but then of course he came on board as a collaborator, and
I'm going to tell you, I looked up a few review papers just to refresh me on the TGF-beta signaling, and they still say endocrine is only found in endothelial cells. So we have a little bit of work to do.
Yeah. Okay. Thank you so much.
Thank you, Dr. Philip. We have to cut it short because we need to get into the next session. So I know that Faxon had his hand up before Kai did. So one last quick question. I'm sorry.
It's okay.
One last quick session and then we'll move on. One last quick question and we'll move to the next session.
Yes, so Rita, that's wonderful, absolutely excellent. I was at the edge of my seat. Sorry, it's not a quick question, I'm just excited. So, so did you compare to microfracture? Because I say that because since microfracture is currently used in the clinic for treatment of osteochondral lesions, it would be nice to demonstrate better hyaline cartilage regeneration compared to the standard of care, which is microfracture as a control. Because for the FDA, it's called the minimum threshold for FDA approval.
So for this rabbit study that we're doing, we used as a standard of care the osteochondral transfer.
Okay.
It might be very interesting to do microfracture and our construct as well.
Yes.
Absolutely. But what we found in the past was that the microfracture, it's a fibrocartilaginous type of repair.
Yes, yeah. Just side by side. I think that's how the FDA likes it. Yeah. Wonderful. Thanks.
Thank you very much for your kind comments, Faxon.
Okay, thank you, Faxon. Thank you, Dr. Kandel, for that wonderful talk and discussion. We're going to move on now to student talks. And first up, we have Matthew Zakaria. He's an MSc student, and his talk is titled Facilitated Fracture Repair via Non-Invasive Localized Cold Therapy. Okay, welcome, Matthew.
Alrighty. Let's see, can everybody see my screen? All right, just double check. Perfect. Alrighty. So let's begin. So hello, my name is Matthew Zakaria, and today I'll be presenting my project regarding the usage of bone therapy within a bone injury site. Briefly, I would like to take a moment just to look back and reflect on the overall development of the project. Within specific populations, a percentage of patients experience a delay in fracture healing or even complete healing failure. This can be attributed to comorbidities and the severity of the injury as it complicates the healing process. One way to counter this is by the utilization of autografts. However, when it comes to the usage of autografts, there is a limited supply. As such, other alternatives need to be considered. However, there are conflicting degrees of success and failure with presented alternatives that we have. Therefore, another solution is essentially needed. Subsequently, conducting a literary review revealed that localized cold exposure through fluids has shown to lead to bone overgrowth with pigs and humans. This made us think about the effects of localized cold exposure treatment on a bone injury site. As such, this formed the basis of a preceding study done within our lab on a cortical defect model within mice. The results attained showed that there was an increase in bone volume and a decrease in ALP expression. Now, the next logical step was to incorporate this treatment into a clinically relevant model. This is where we decided to utilize a closed femoral fracture model to mimic clinical scenarios. As such, our objective was to examine the anatomical and cellular effects of cold therapy on a fracture in a clinically relevant model. Consequently, our hypothesis is that enhanced bone healing demonstrated using cold therapy in cortical defects will be replicated within a closed fracture healing model and will better approximate human clinical situations. Now, the methodology involved the creation of a bilateral closed femoral fracture model within mice. Firstly, a guide wire was inserted, followed by the usage of a blunt guillotine driven by a drop weight in order to mimic high impact fractures. The fractures were then stabilized by the incorporation of an intramedullary pin. The guide wire was then removed. Following the surgery, the mice were then separated into 3 groups regarding the total duration of the treatment. One group was treated for 28 days, another for 14 days, and another for 7 days. Now, the treatment involved daily cold therapy where the randomized experimental hind leg was exposed for 15 minutes in a cold water bath to reach an internal bone temperature of 19 degrees Celsius. Following the completion of the treatment for each group, the femurs were then harvested and micro-CT and histological staining analysis was conducted. Now, within the 28-day group, there was a significant increase in bone volume and trabecular thickness, as well as a significant decrease in trabecular separation, signifying that there was facilitation of the fracture repair process. Now, interestingly, the 28-day group demonstrated a significant decrease in ALP staining of osteoblasts, while levels of trap staining of osteoclasts stayed stagnant, indicating that the callus had begun to undergo remodeling. Additionally, within the 28-day group, vasculature parameters showed that the vasculature network was enhanced as channel volume over tissue volume and channel thickness increased, while channel separation decreased. Now, the 14-day group displayed similar findings to the 28-day group in that there was a significant increase in bone volume and trabecular thickness, as well as a significant decrease in trabecular separation, signifying that there was a facilitation of the fracture repair process as early as 14 days. Now, the results attained confirmed that localized cold therapy accelerates bone repair within a clinically relevant closed femoral fracture model. It confirms our hypothesis in that the findings from a cortical defect model can translate to a clinically relevant femoral fracture model. Now, the results attained demonstrate that osteoclastic-driven bone remodeling is attained faster within cold-treated femora. Additionally, analysis of the bone channels indicate that the vasculature network within the injury site is improved following cold therapy. As such, cold therapy has been shown to increase bone tissue, improve the vasculature network within the injury site, and lead to favorable microenvironment fluctuations regarding certain biomarkers of bone cells. As such, this leads us to conclude that fracture repair is accelerated by the incorporation of localized cold therapy. Lastly, I would like to thank my fellow colleagues as well as the organizers of this event for the opportunity to present. Thank you.
Thank you, Matthew. I'll open this up for questions from the audience. Time for one quick question. Okay, I'll ask you one quick, Oliver, you had a question?
We're leaving the questions at the end of the sessions.
We leave the questions all the way to the end of the sessions. Okay, now that's well noted. Okay, and so, okay, we'll move to the next speaker. And the next speaker is Oliver Wu Martinez. He's an MSc student, and his talk will be evaluation of synergistic effect of senolytic combination treatment for intervertebral disc degeneration.
Thank you, Dr. Rosenzweig. So low back pain is experienced by 80% of the world's population. It's associated with intervertebral disc degeneration. So as you can see in the middle image, we have a young, healthy donor. And so you can see there's no discoloration. It has very elastic properties as well as high levels of proteoglycan, and for that reason has high water retention. But because of age, injury, and even genetic factors, you can actually start losing some of these properties and developing degeneration, shown here. So this is a very extreme case, but this is actually a donor that we've received at the hospital. And intervertebral disc degeneration has been linked with an increased number of senescent cells. So what are senescent cells? Well, senescent cells are cells that no longer proliferate, but are not considered quiescent. So these, and for that reason, it's because they can no longer revert back to a proliferative state. They adopt a senescent associated secretory phenotype, or SAS for short. And so this release of cytokines, chemokines, as well as nociceptive factors can further cause the degradation of macromolecules, inflammation, as well as pain because of the adjacency to the spinal cord. And our lab has also shown that PAM2CSK4 further induces senescence by activating toll-like 2 receptors. So one way to start removing some of these senescent cells is the use of senolytic drugs. So these reduce SAS factors as well as actively remove them via apoptosis. And so our lab has been using a natural senolytic, ovanillin, which also has anti-inflammatory properties, and a synthetic senolytic, RG7112, which is a pure senolytic. So we hope that by doing so, we can restore some of that balance. And so we wanted to first look at different physiological models to test out these senolytics. So we're very fortunate at the Montreal General to have access to surgical samples from patients undergoing back surgeries for back pain, as well as spine donors. So first we screened senolytics in the monolayer and pellet models, and then we moved to ex vivo experiments. But the issue with the monolayer pellet as well as the IVD is that we can measure senescence, we can measure the different levels of SASPs, but we can't actually measure pain behavior itself. And that's where the murine model actually comes in. And so we decided to use spark null knockout mice, which around 6 months starts developing degenerative degeneration, mimicking human intervertebral disc degeneration. So the first set of experiments was really to figure out the combinations of ovanillin and RG. So we ran serial dilutions of both, selected different concentrations, and made combinations of both of them. So as you can see here, we're looking at the senescent levels in pellets. So we can see that control, which was actually PAM-induced, and everything was PAM-induced, we can see that the single treatment is able to reduce that level of senescence. But when we combine the two senolytics, we can see that we can actually further reduce the amount of senescence in these pellets using the P16 as our marker within IHC immunofluorescence. For the animal models, we used 3 different methods to evaluate pain behaviors. So the first was grip strength, which evaluates axial discomfort. And the next was the von Frey filament, as well as the acetone test to measure radiating pain. So what we did was we treated the mice every week, and we evaluated in this case, the grip strength every 2 weeks. So as you can see with the baseline, there's a discrepancy between the spark null mice and the wild type. But as we progress every 2 weeks when we're doing those grips tests, we can see that there's further loss of grip strength, hinting that there is a continuous degeneration of the spine. But when we're treating with the single drugs, you can see that we're actually able to inhibit that degeneration and improve the grip strength just a little bit. But what's really interesting here is when you look at the combination treatment, we're actually able to not only inhibit that degeneration, as you can see in the SPARK cohort, but in the combination, we're actually able to bring it up to wild type, which was very, very exciting. And so after the in vivo studies, we actually cultured the IVDs from the mice and looked at the protein expression from the media. So we can see that we ran a plate of 15 different proteins, and out of 10, the senolytics was able to actually improve the SAS factors. So here we have, as an example, IL-1B, which is a cytokine, and CXCL5, which is a chemokine. So when we are looking at the wild type and SPARK, we can see there's a large discrepancy. But when treating with the single drugs, we're able to actually bring that down twofold. And then again, with the combination shown in green, we're actually able to bring that down twofold from the single drugs. So we're really able to see this kind of additive and almost synergistic effect of the combination. So our lab has been able to show that there's species compatibility for use of these two senolytics, as well as that the combination has a greater ability of removal of senescent cells, as well as improving the behavior signs in vivo, as well as reducing the SAS factors in vitro and in vivo. So senolytics and really the combination of these two senolytics has great potential for treating low back pain. There's still a lot of ongoing work, such as the assessment of degeneration in spine, better understanding the different pathways of the combination treatments, as we know that ovanillin treats we've shown that it treats 3 different pathways, and then RG goes through another pathway, but when combining them, is there maybe another pathway that we're actually targeting, as well as evaluating the pain and senescent marker in both the spinal cord and spines following treatment. So senolytics, they work in monolayer, in pellets, in ex vivo models, in vivo. So hopefully maybe one day we'll be able to create a treatment for patients with low back pain and maybe prevent or even revert disc degeneration. So So I'd like to acknowledge my lab as well as my PI, Dr. Haglund, the spine teams, and all the donors, as well as our collaborators at the Stone Pain Lab. Thank you very much.
Thank you for that nice talk, Oliver. We're going to move to the next presenter, Hao Yu Liao. I hope I said that correctly. MSc student, regulation of lysophosphatidic acid signaling in human spermatozoa.
Can everyone see my screen?
Yes.
Okay. So let me start. Hi, everyone. My name is Hao Yu Liao, and my supervisor is Dr. Christiane Overherty. Today, I'm going to talk about my project entitled Lysophosphatidic acid signaling in human sperm viability. Firstly, we should know why the sperm viability is important, because spermatozoa must reside 6 to 8 hours in oviduct in order to acquire the ability of fertilize the oocyte. Therefore, it is essential that the sperm remain viable during this time. Okay. From the previous study in our lab, we demonstrate that the inhibition of the PRDX6 phospholipase A2 with MJ33 impaired the sperm viability by preventing the phosphorylation of the PI3 kinase and the AKT substrate. However, the presence of the active LPA signaling pathway in mature spermatozoa is unknown. So our hypothesis in this project is that LPA signaling is active in spermatozoa and regulated by different kinases. And there are two objectives. The first one is to determine the presence of LPA receptor in human spermatozoa and whether or not the LPA signaling would activate the downstream PI3 kinase and the AKT pathway. And second, to study the it could be regulated by different kinases or not. Okay, here is our method. The highly motile sperm from the human semen was used to localize the LPA receptor and also study whether or not the LPA pathway is active by specific inhibitor. The result in here shows the LPA receptor 1 and 6 are localized in the tritone soluble fraction, and the LPA receptor 1 also could show the signal in the insoluble fraction of the human spermatozoa. In the immunocytochemistry result, 4 different LPA receptors showed in tail and midpiece in the non-permeabilized sperm. And the LPA receptor 1 also has the signal in the post-echosomal region and the echosome region in the permeabilized sperm. Otherwise, and also the LPA receptor 3 and 5 signal showed in the equatorial segment and the echosome region, respectively. And using the LPA receptor Ki161425, we found the inhibition of the LPA receptor 1 to 3 would decrease the sperm viability and phosphorylation of the PI3 kinase and the AKT substrate as well. Then, using the different kinase inhibitor, demonstrate that the calrithrin and the TA47, which is the inhibitor of the PKC and the receptor tyrosine kinase, lead the sperm death and also prevent the phosphorylation of the PI3 kinase and AKT substrate. Furthermore, studying whether the phospholipase C, the upstream of the PKC, is active in the signaling, and we found the phospholipase C inhibitor affect the sperm viability, but could rescue by the exogenous deacylglycerol. So our conclusion in here is LPA receptors are active in the human spermatozoa, and the PI3 kinase and AKT pathway is regulated by the LPA receptor, phospholipase C, PKC, and the receptor tyrosine kinase to maintain the sperm viability, as you can see in this scheme. And this research will help us to decipher the molecular mechanism to design the novel treatment to maintain the sperm viability in infertile men. Thanks for your attention, and thanks those lab member, collaborator, and who support this project.
Thank you very much, Hao Yu. We'll go to the next speaker, and that is Wala Alamadi. And the talk is Precision Nutrition Increases Efficacy of DNA Damaging Therapies in Prostate Cancer. voila, you can go ahead.
Yes. Hi, everyone. Thank you for giving me the opportunity to present part of my project today. My name is Wala, and I'm a PhD student at Dr. Lebey's lab. I'm here today to present part of my PhD project, which focuses on the use of precision nutrition to increase the efficacy of DNA-damaging therapies in prostate cancer. Now, prostate cancer is the most common cancer among Canadian men, and it's the third leading cause of death from cancer in men in Canada. And it was estimated in 2021 that prostate cancer diagnosis would represent 20% of all new cancer cases and 10% of all cancer death in men. There are several risk factors that have been linked to prostate cancer, and my project focuses on the dietary part, where data from 2 human cohorts have looked into the link between the fat intake and prostate cancer. They found that each additional 5% increase in the total calorie intake from saturated fat is associated with 2.8-fold excess risk of prostate cancer mortality and 81% greater risk of overall mortality. And replacing 10% of the energy intake with vegetable fat was associated with lower risk of all-cause mortality. And this indicates that a specific type of fat intake has a direct effect on prostate cancer progression. Now, our data shows that in MYC-driven prostate cancer mouse model, chronic high-fat diet feeding that lead to diet-induced obesity resulted in increase in DNA damage in MYC-transformed prostate, as shown over here by the gene set enrichment analysis showing enrichment of gene sets related to DNA repair, and also shows elevation of the level of gamma H2X in Western blot in the tumors and by immunofluorescence staining. And by using MYC cells derived from that high MYC genetically engineered mouse model allografted in FBB mice, we were also able to observe the induction of DNA damage in chronic high-fat diet feeding, as well as in short high-fat diet feeding. So we asked the question, could a short-term saturated fat diet increases prostate cancer sensitivity to DNA damaging therapies? And to answer that question, we designed an in vivo experiment where ionizing radiation was used as a DNA damaging treatment to determine the ability of the high-fat diet-induced DNA damage to sensitize the tumors. And we used MYC cells allografted in FEB mice. At the enrollment, all mice were on controlled diet and injected with MYC cells while being on controlled diet. And once tumors are palpable, mice were randomized to continue on controlled diet or to be switched to high-fat diet feeding. Ionizing radiation was given when the tumor size was between 79 to 113 millimeter cubic, and the duration of the high-fat diet feeding was a minimum of 2 days and a maximum of 8 days prior to ionizing radiation, and all the mice on the high-fat diet arm were kept on an additional 2 days of high-fat diet feeding post-ionizing radiation to maximize the DNA damaging effect. Altogether, high-fat dietary intervention lasted between 4 to 6 days, and we observed that irradiating the tumors while on high-fat diet significantly extended the tumor doubling time. So we asked the question, what is the diet component that derives the DNA damage induction? And to answer that question, partially, we designed a hybrid in vivo in vitro experiment where we collected plasma from tumor-free mice that were put on a 10 days high-fat diet or a 10 days control diet. And we used the pool of the plasma collected from each diet group to treat MYC cells in vitro. And we showed that high-fat diet-associated circulatory factors recapitulate the induction of gamma H2X observed in vivo and induce accumulation of DNA double-strand breaks, as we can see over here by IF staining, and also by quantifying the tail moments by Comet assay. Finally, I want to end my presentation with this slide where we wanted to evaluate the translation potential of our preclinical results. So we analyzed the dietary and the transcriptomic data available from 2 human cohorts, the HPFS and PHS cohorts, and we found that in men with localized prostate cancer tumors, a DNA repair transcriptional signature is enriched by saturated fat intake, possibly in response to greater DNA damage load. So we hypothesized that saturated fat intake might also expose vulnerability that could be exploited therapeutically through precision nutrition strategies. Thank you very much for listening. I also would like to thank my supervisor, my lab members, collaborators, and the funding agency. Thank you.
Thank you, Wala. Okay, so now I'll open up the panel discussion for questions to all our presenters. We have about, according to the timing, we have 4 minutes left, but I think we can maybe squeeze a few more questions in. Okay, Dr. Kandel, first question is yours.
Thank you very much to all the presenters. They were great studies. I think that I start with a question for Oliver. I was intrigued about the spark null mouse and the degeneration. What is the mechanism that leads to increased senescence of the cells?
So basically senescence is a protective mechanism, but a large part of the time, so there's different pathways at which a cell can become senescent. So telomere reductions, as well as different cytokine. If there's too much inflammation, it is a protective mechanism. And as well as genetic, but I'm not entirely sure if I'm fully answering your question properly, or like maybe I'm giving too much of a generic answer.
No, that's a great answer. Is the increased senescence in some way related to the spark null phenotype or genotype?
So basically what we did was we showed initially that toll-like-2 receptor activation does increase senescence. And in part ways, it can be an indirect way of senescence. So through the inflammatory cytokines and things like that, it can induce senescence, but also it can actively do it. But the spark null mice is something completely different. So basically, spark is a type of protein that is very acidic and cysteine rich. And so a loss of that can cause degeneration within the intervertebral disc. So it's two different things from the senescent induction that we did, and then the spark null.
Okay, great, thank you. So those drugs would work generically across both then?
That's what the data says. Exactly.
That's great, thank you.
Thank you.
Hey, Dr. Mwale.
Oliver, very interesting work. I know that if you take senescent cells and you inject them in a joint, that's been published, you get osteoarthritis. So question one, has it been shown that if you inject them in the disc, you get disc degeneration?
From what I know, I haven't read anything of that sort, but I would assume that they would, because again, going back to the fact that senescent cells do have a SASP phenotype, we would assume that because of that reason, you're going to have an increase in cytokines and chemokines and very inflammatory environment, which would affect the tissue milieu.
Okay. So my other question is, why not just use a SASP inhibitor? There's a SASP inhibitor called roxolitinib, which is a Janus kinase inhibitor.
So I think that would be maybe a little bit too specific. That's why the use of ovanillin is actually beneficiary, because we're able to remove the senescent cells, but it also has anti-inflammatory properties. So we're able to reduce that level of SASP while targeting the cells that are actually producing those kinds of inflammatory mediators.
Okay, thanks.
Okay, thank you. Do we have more questions from the panel?
Yeah, I have quick questions, actually. One, sorry for ganging up on you, Oliver. Just a question on the senolytics. So your treatment in the spark null mice, it was progressive, right? Or continuous throughout the development of the pain model, correct?
Exactly.
So have you looked into whether if you treat the animal or a model, if you have an animal model for this or not, but have you looked into the fact of treating already established pain and seeing how senolytics affect like an animal that already has established some sort of discogenic pain, if it would reverse it?
So right now we do have some ongoing animal studies. So one is starting from a prevention method. Mm-hmm. So we're actually treating these younger mice and seeing how they progress with degeneration. The other one is, in this case, they were 6 to 7 month old mice is not necessarily young, but it's not necessarily old. So that is the method that we did for this experiment. But then the next set of experiments is actually getting much older mice and treating it and see if we're able to actually revert it back.
Mm-hmm. Perfect. Next question is for Wala. Very interesting, actually, the relation between saturated fat and sensitivity to radiotherapy. was just wondering, how would you translate that to the clinic? So how would you give that to a patient? What type of therapy would you give in terms of saturated fat to a patient?
So that's a very interesting question. One of the other experiments that we did is we did a lipidomic study where we wanted to know what a specific type of fat or saturated fat in the plasma that is inducing this effect of DNA damage. And we have like a panel of lipids that were enriched in the high-fat diet and in the diet group and were not enriched in the control diet. And at this point, we're testing those lipids, and we're going to proceed with in vivo experiments where we can use selective lipids and inject them in mice in the plasma. And then if we can reproduce our experiment, this would be a translational potential to the clinic, yes.
I have a question, sorry, as a follow-up to this. You're saying that high-fat diet actually makes the cancer radiosensitive or resistant?
Radiosensitive.
I see. And it's the saturated fats.
Yes, yes. As we observed that saturated fat induces DNA double-strand break, and a lot of cancer treatment uses like a combination therapy to enhance the DNA damaging effect and eventually kill the cancer cells. So we are trying to exploit this advantage of the saturated fat to induce DNA double-strand break to eventually, for example, deliver a lower dose of radiation to produce a better effect in response to therapy.
Okay.
Thank you. Dr. Mwale had his hand up again.
No, it's an order.
Okay. So I think we have about 2 more minutes left for questions, according to Matthew. Anybody else from the audience? I have one question for Matthew. You had some nice data on the bone repair following the cold therapy.
Yeah.
And you showed a lot of nice histology with the remodeling. And it's a 4 weeks study, correct?
Yes. So, to be exact, the mice were divided into 3 groups. What's called, one was 28 days, one was 14 days, and one was 7 days. The results that I showed that were relevant were the 28 days and the 14 days. For the 7 days, the data that was obtained for the bone was, it was not significant, and therefore, for time reasons, I just elected to not really include it.
Did you consider testing the mechanics? Because it looked like on some of the samples you showed, it had nice cortical repair already at 4 weeks.
We did not. We did not. Yeah.
Okay. Other questions from the audience? No? Okay. Then we have an announcement from Dr. Berlet.
Thank you very much, Derek. Appreciate it. Do you want to thank the people in your session before it's not related to your topic, so I guess that's the end of that session and the research day.
That is the end. Sorry, that our time is up now. Yes.
Yeah. So it's very rare that we all meet. Extremely rare, and even rarer than it used to be. So I'm sorry to take the time to make an announcement. Some of you are in the injury repair and recovery program at the RI. Some of you are interested in innovation. If that's not you, then you can phase out for a couple of minutes before Dr. Philip announces the winners today. But I'd just like to announce that the IRR and the CLIP has a new program manager, Mathilde Guibert. She joined a couple of weeks ago. She's had positions marketing for several industries in France, Netherlands, and Spain. She has a master's in economics and management, and a bachelor's in economics and management, and a master's in international business and management. Mathilde, if you're here, maybe you could just turn your camera on and give a wave so people can recognize you. Mathilde is located on C9 at the Montreal General Hospital. Could you say a few words, Mathilde?
Yeah, so hi everyone, and thank you, Jake, for this fantastic presentation. I always take this opportunity to congratulate and to thank all the presenters for today. So as Jake mentioned, I'm the new program manager for the Injury Repair and Recovery program, and also joining the CLIP, so the Clinical Innovation platform. It's as it said that you can find me there. So if you want to have a chat or if you have any questions, feel free to reach me. I will also indicate my email address on the chat, so don't hesitate. I joined on a few words about me. Indeed, Montreal a few weeks ago. I come from France, and indeed I had several experiences, and I was previously in a tech company because I was really looking to be in this innovative environment. And as I wanted to have more information and more knowledge on innovative healthcare, I was looking forward to join the research institute. So I'm really pleased to be in this new environment and family. So thank you for welcoming me. And thank you again, Jake, for your introduction.
Brilliant. Congratulations again on joining us, and look forward to working with you. Okay, thank you very much. I'll pass you back to Dr. Rosenzweig.
Thank you, Dr. Bartlett. So that closes our session for this afternoon, and I wanted to thank all of the speakers, including Dr. Kandel, and all the students for very nice presentations and a lot of nice work shown. So I'm going to pass it, I guess, now to Dr. Philip, who will be taking the awards for this session. Is that correct, Faxon?
Yes.
So
Annie?
Thank you, Derek and Faxon. Now we have the moment that the trainees are anxiously waiting for, the announcement of the awards. Let me take this opportunity to congratulate all the presenters. I was really impressed by the high caliber of all the presentations. So congratulations to all to making this huge effort and presenting your best work. The scores were very close to each other, and it was tough to pick the top ones. Each of the sessions were scored separately, and so there are a prize for each session. And again, congratulations to all the presenters and the winners. So I'll announce the winners right now. For session 1, it's Claudia Covarubas from the San Campo lab. For session 2, can you hear me okay?
Yes, we can, Annie.
Okay, for session 2, it's Makina Pook from Giulio Fiore lab. And session 3, it should be, it's a typo. Oliver Wu Martinez from Lisbeth Haglund lab. And for the poster presentations, breakout room 1, the winner is Sabrina Leo from John Shervenkopf lab. And for the breakout room 2, orthopedics presentations, it's Dagi Gowell from Derek Rosenweg lab. And for breakout room 3, clinical outcomes, it's Said Asser from Jason Harley lab. And breakout session room 4, urology, gynecology, Michelle Shen from David Labelle lab. So congratulations to all the winners, and I should congratulate all the presenters as well, who have done a tremendous job of showcasing their results, and excellent work. Thank you all.
Thank you, Dr. Philip. Congratulations to all of the presenters for their awards, and I'll pass it back to Dr. Mwale.
Yeah, thank you very much, everyone. I think all the organizers who helped to keep this where it is. I think Dr. Bartlett in the beginning talked about, mentioned all the people who were involved in organizing. And one thing that we would like is for you to send us your comments, any improvements we can make so that we can adjust for next year's research day. Congratulations to everyone, and I would like to thank the keynote speakers. Really wonderful presentations, and everyone gave really great presentations. And please have a wonderful summer, and thank you very much. So, bye everyone.
Thank you very much.
Yeah.
Bye everyone.
Thank you.
Bye-bye.